titled Advances in Experimental Therapeutics, being held here at the Center of Advanced Studies in Chemistry, Panjab University, Chandigarh, from July 7 to July 11. This course, organized under the Global Initiative of Academic Networks, GIAN, brings together global and local expertise to explore next-generation therapeutic strategies. Before we begin the workshop, I would like to request all of you to please stand up for Panjab University anthem.
तमसो मा ज्योतिर्गमय तमसो मा ज्योतिर्गमय तमसो मा ज्योतिर्गमय तमसो मा ज्योतिर्गमय
पंजाब विश्वविद्यालय तेरी शान और शौकत सदा रहे, मन में तेरा आदर मान और मोहब्बत सदा रहे। पंजाब विश्वविद्यालय तेरी शान और शौकत सदा रहे। तू है अपना भविष्य विधाता, पंख बिना परवान सिखाता, जीवन पुस्तक रोज पढ़ा कर सही गलत की समझ बढ़ाता।
जीवन पुस्तक रोज पढ़ाकर सही गलत की समझ बढ़ाता।
तेरी जय का शंख बजाए,
रोशन तारे बन जाए,
बखरी तेरी शोहरत, तेरी शोहरत सदा सदा रहे। विश्वविद्यालय तेरी शान और शौकत सदा रहे। पंजाब विश्वविद्यालय तेरी शान और शौकत सदा रहे।
तमसो मा ज्योतिर्गमय तमसो मा ज्योतिर्गमय।
Thank you. Please be seated, everyone. Now, to begin, I would like to request Professor Sanjay V. Malhotra to extend a warm welcome to Professor Y.P. Verma, Registrar, Panjab University, Chandigarh, by presenting him with a bouquet of flowers. I request Professor Gangaram Chaudhary to warmly welcome Professor Meenakshi Goyal, Director RDC, Panjab University, Chandigarh, by presenting her with a bouquet of flowers. I request Professor Y.P. Verma to welcome Professor GRC with bouquet of flowers. Okay, so sorry. I request Professor GRC to welcome Professor Y.P. Verma with bouquet of flowers. I now request Professor Gurjaspreet Singh, Coordinator of GIAN Program, and Dr. Aman Bhalla to extend a heartfelt welcome to Professor GRC with bouquet of flowers. As we begin this auspicious event, let us invoke the blessings of knowledge and wisdom. The lighting of the lamp symbolizes the dispelling of darkness and the ushering in of enlightenment and knowledge. I would like to request our esteemed guests to join us in lighting the ceremonial lamp. Let the light guide us towards the path of innovation and discovery. To formally inaugurate this workshop, I would now like to request Professor Gurjaspreet Singh, Department of Chemistry, Panjab University and local coordinator of GIAN, to deliver the welcome address.
A very good morning to Professor Y.P. Verma, Registrar, Panjab University, Professor Meenakshi Goyal, Director, RDC, Professor Gangaram Chaudhary, Chairperson, Department of Chemistry, our worthy speaker, Dr. Sanjay B. Malhotra, a Professor at Department of Cell Department and Cancer Biology at Oregon Health Science University, Oregon, USA. Course Coordinator, Dr. Aman Bhalla, faculty members, all the participants, and everyone present here. I welcome you all to International Workshop on Advances in Experimental Therapeutics, an event under Global Initiative of Academic Network GIAN. Dr. Malhotra obtained a PhD degree in chemistry and trained under Nobel laureate Professor Herbert C. Brown at Purdue University. He is also a Fulbright specialist and fellow of Royal Society of Chemistry, UK. His expertise in medicinal chemistry has led to the preclinical and clinical advancement of drug candidates. Global Initiative of Academic Network GIAN in higher education was launched in 2015. It is a program of education aimed at tapping the talent pool of scientists and entrepreneurs internationally to encourage their engagement with the Institute of Higher Education in India. GIAN envisages to catalyze higher education institutions in country, and it includes all IITs, IIMs, Central Universities, IISc Bangalore, IISERs, NITs, IIITs, subsequently covering good state university where the spin-off is vast. GIAN is an evolving scheme which includes participation of foreign faculty in institute as visiting faculty or professor of practice to participate in delivering 5 or 10 days courses. The first course was launched by Panjab University in year 2016. 25 courses have been conducted so far, and we are looking forward to conduct more courses in future. Currently, 10 courses have been sanctioned in phase 4, that is in 2024 to 25. The call for the fifth phase will be released shortly. Our motive is to engage more and more participants from various institutes and to accelerate the vigor of GIAN. For further detail, you are most welcome to contact me regarding the GIAN. I wish you all a great learning experience with Professor Sanjay Malhotra. Thank you.
Thank you, sir, for your kind words and for setting the stage for what promises to be a highly insightful workshop. We are deeply honored to have Professor Y.P. Verma, Registrar of Panjab University, in our midst today to share his thoughts and grace the occasion with his words.
Professor Malhotra, our esteemed speaker for the GIAN program, Professor Meenakshi Goyal, Director Research and Development, Panjab University, Dr. Gujaspreet, Dr. Gangaram Chaudhary, Chairperson of Department of Chemistry, and Dr. Balla, all colleagues, students, and the researchers from different institutions. My greetings to all of you, and it's indeed a privilege to be here today. On this occasion of inaugural of the GIAN program, it's a wonderful program by Government of India, where the faculty from the foreign is called to the institutions in India, and he will share his best of his research and expertise with the students of our institutions. This program, I was also one of the part of one Vajra program where we used to call faculty from Indian origin from outside to stay here in India for 2 months time, and it used to be a very wonderful experience, reason being that the experience, the level of research that they carry out, it is directly brought into the classroom straight away. So in today's time, when we say that we talk about a globalized world, the globalization has taken place, and it is very important to know that what is going on in the surrounding universities, and institutions, and maybe at different level of research, and then only we need to prepare. I must say that when you talk about India, as of now, India is progressing very well. We already are the 4th largest economy, and the programs like this is not simply just to show that we are calling international faculty, but it's, I think, it is a larger interest and larger goal behind that. The goal which I feel is that we need to prepare ourselves to be in the top 1 or 2 economy in the time to come. We are already envisioning the Viksit Bharat by 2047. So it will not be simply that we give a slogan and it will become automatically the Viksit Bharat and we are going to be among top 2 economy in the world in the time to come. What are you going to do behind that? You just need to understand the way China has progressed, the way they have done wonderfully well as far as education is concerned, the research is concerned, the manufacturing is concerned. We are also targeting certain areas, but what we lack perhaps is the kind of exposure or the zeal, I should say, to compete with those countries like China. Why we need to? Because sometimes we also need to admire the best practices of the world. Obviously, we cannot talk about the US, there is a different culture altogether, but at least when we compare ourselves with the China, so at least we can say that we were at the same level and where they are now and why we are not at this level. So it's a time for people like you where we need to pull up our socks now, get ready, gear up if you really want to compete with the world in the time to come. Why do I say this is because of the reason that these little exposure that we get from these faculty, I think when I used to study, we read 1 or 2, equations. One was your Jha transformation, and it looks so wonderful. कि there is a Jha, somebody from India, उसके नाम पे transformation है. Otherwise तो हम Rimmer-Tamer rules, आओ पाओ rule ही पढ़ते हैं. So can't we develop things like that? Can't we go to the research level where we also have certain nomenclature, certain formula, certain reactions on our name? That should be the aim. And chemistry have a big role to play. Why do I say this? Because the reason that you must have seen that India is now targeting the semiconductor technology, where we are all feeling proud that we are the, I think, second largest manufacturer of iPhone. But do you know from where the chips come, from where the technology and everything comes? We are simply assembling it. Why can't we manufacture it? And when it comes to competing with the world mobiles, I think we hardly have any mobile over here. Motorola cannot compete with that. So I think these are the things, and chemistry people have a big role to play in that. When it comes to industrialization, the manufacturing sector which need to grow up, it has a big role to play. In semiconductor technology also it has a big role to play. So there are so many areas. In environment sciences you have a big role to play. So I think what is more important is that get the idea from the people from outside. No doubt these days we have access from whole world. We know what's going on in and around. You can get the access to the paper. But what is advantage of getting faculty over here is that whatever research that he must have carried out in the last 20 years, you can really directly ask. From papers you can get few things, but you can get the clarification. So those clarification I think you can directly get from here. And it is also important that somebody who is coming here to deliver talk, you should go through his research also. If it interests you, it's of your area of research or interest, go through those papers. Be ready with the questions. Ask, make best use of the time, 7 days time that you get over here. And that's how I think you can reach that maybe your the coveted goal that you set for yourself. I really feel that I cannot say that each and every one of you can be a very good researcher. Everyone has their own potential, but there are few, 1 or 2 out of you who can really deliver, who can really come up with those sort of things which I am talking about, certain reactions in your name, certain some innovative work in your name, and that is what you need to target. So this is such a initiative by Government of India. I think there are lots of money being spent on these programs. Let's make best use of this. At least try to inculcate this culture of research and innovation, your next generation. Somebody will definitely, with the passage of time, things are not done overnight. It takes time, but it's very important that the ecosystem, the culture that we create. Right now, I think you go to one step, then you go to the next step. Obviously, when we pass, we go from class 1 to class 12th or the colleges. So this is the building step for all of us. We have to go to PhD and postdoc in the research area. So let's start building. Do it sincerely, pass on a culture and a environment to our juniors with this sort of mindset. Yes, we really need to deliver. We are the best and we can deliver, and tomorrow we have to be the world leader. With this particular motive, I feel that this program should really ignite your instinct to do the best, and I wish all the best to you for the deliberation that will take for the next 7 days over here. And I would like to personally thank Professor Malhotra for taking out time for the students, for the Panjab University, sir. We welcome you from core of our heart, and I feel your expertise is definitely going to help our students, and there must be some collaboration also, but collaboration will take place only if, what I said, if you read his paper, you just cannot go blank and say कि मुझे कुछ वहाँ पे PDF करना है या करना है. You need to पहले पात्र बनो, बाकी सफलता खुद मिल जाएगी, opportunity भी मिल जाएगी आपके साथ. So read those. Make yourself capable. I think opportunity will knock at your door. So thank you so much, and best wishes to all of you.
Thank you, sir, for your valuable insights and for always supporting academic endeavors like this one. We are privileged to have Professor Meenakshi Goyal with us to deliver the address.
Esteemed faculty members, distinguished speaker, dear participants and students, wish you all a very pleasant morning. It gives me a great pleasure to welcome you all to this prestigious GIAN course on Advances in Experimental Therapeutics hosted by the Department of Chemistry. This course marks a significant academic milestone, one that brings together global expertise, cutting-edge science, and a shared commitment to innovation and knowledge dissemination. At the Research and Development Cell, We remain steadfast in our mission to cultivate a thriving environment of research excellence, interdisciplinary synergy, and global academic engagements. The GIAN program, that is Global Initiative of Academic Network, an initiative of the Government of India, epitomizes this spirit by facilitating the exchange of knowledge between eminent international scholars and the Indian academic community. We are especially honored to host Professor Sanjay V. Malhotra, an internationally recognized leader in medicinal chemistry and cancer therapeutics. His pioneering work in drug development and translational research continues to shape the landscape of biomedical science. Professor Malhotra's presence here is not only a privilege, but a profound opportunity for all of us to learn from one of the finest minds in the field. The theme of this course, Experimental Therapeutics, is both timely and transformative. As a multidisciplinary domain, it encompasses the development of innovative treatment modalities, including small molecule drugs, biological, gene, and RNA therapies, and nanomedicine. What sets this field apart is its traditional focus, its ability to bridge the divide between fundamental discoveries and real-world clinical application. In a world confronting escalation challenges such as drug resistance, emerging diseases, and complex conditions like cancer and neurodegeneration, the need for targeting and effective therapies has never been more critical. Thanks to advancements in computational design, molecular biology, and high throughput technologies, we are witnessing a redefinition of the drug development paradigm, faster, smarter, and more precise. This course offers a rare opportunity for students, researchers, and faculty to delve into the most current strategies, technologies, and challenges in experimental therapeutics. It is not just a training module, but a platform for dialogue, idea exchange, and potential collaboration. I would like to congratulate Professor Gurjasprit Singh and his team for their unwavering commitment and academic vision in organizing this course. Bringing together leading minds from India and abroad is a remarkable accomplishment, one that speaks to the department's leadership and dedication to excellence. To our distinguished speaker, thank you for your generosity in sharing your knowledge and experiences. I would like to say to my participants, immerse yourselves fully in this learning journey, ask questions, engage critically, and take back insights that will shape your research and professional pursuits. This GIAN course reaffirms our university's dedication to promoting globally relevant research and empowering the next generation of scientists and innovators. Wishing this program resounding success and a catalyst for future innovations. Thank you.
Thank you so much, ma'am, for your wonderful thoughts. To conclude this inaugural session, I now invite Dr. Aman Bhalla, Department of Chemistry, Panjab University, to deliver and introduce the theme of workshop and also deliver the vote of thanks.
A very good morning to all present. It's my proud privilege to share a few thoughts on the theme of GIAN workshop, that is advances in experimental therapeutics. Experimental therapeutic is one of the most exciting and fast evolving field in the science today. It focuses on discovering and developing new treatments to fight disease, not just by trial and error, but through a deep understanding of biology, chemistry, and molecular medicine. This field lies the intersection of chemical biology, pharmacology, drug design, and molecular diagnostic. It uses advanced tools to study how diseases work at the molecular level and then applies that knowledge to develop target therapies, drugs that are more effective and cause fewer side effects. Today, as a disease becomes more complex and personalized medicine becomes the new norm, the importance of experimental therapeutics cannot be overstated. From platinum and gold based metal complexes in chemotherapy to asymmetric synthesis in drug design and molecular imaging for drug tracking, the techniques covered in this course are shaping the future of how we treat disease. This workshop is designed not only to introduce participants to these cutting edge tools and ideas, but also to inspire them to think in an innovative way. It encourages collaboration across the different disciplines. I believe this workshop offers something valuable to everyone. With that, I now take this opportunity to deliver the vote of thanks. On the behalf of organizing team, I extend our heartfelt thanks to Professor Renu Vig, Vice Chancellor, Panjab University, Professor Y.P. Verma, Registrar, Panjab University, Professor Yojana Rawat, DUI, PU, Professor Meenakshi Goyal, Director Research, Professor Sanjay V. Malhotra, the guest speaker, Professor G.R. Chaudhary, Chairperson, Department of Chemistry, Professor B.S. Murthy, National GIAN Coordinator, and Professor Gurjasprit Singh, Local GIAN Coordinator, and all the participants, organizing team members, teaching and non-teaching staff, and volunteers. Your participation makes events like this meaningful. We are grateful to all the speakers and experts who have taken the time to share their knowledge and experience. Thank you all once again. Wishing you a continued success in your scientific journey. Thank you very much.
With that, ladies and gentlemen, we conclude our inaugural session. Let's take a short break for refreshments. Please join us for high tea and return by 11 a.m. as we begin the first lecture of the workshop.
चाय पी लो, बाहर चाय लो, चाय पी 11 o'clock स्टार्ट कर लेना, चाय पी लो। वो किसका रहेगा? गंगा की है। ये फालतू के साथ ही देते हैं, इसका साथ उनकी गाड़ी की नहीं करता। मित्र, ये इसका उनकी गाड़ी खड़ी है।
है तो 11 है तो 11 यार, बैटरी ड्रेन हो रही है इसकी। तो मैं चार्जिंग पे लगा दूँ इससे वो करूँ? फिर इनसे पूछना था, डिस्कनेक्ट कर दूँ मैं इसे? वो रुको, पूछो तुम। यहाँ मैं देखती हूँ फिर।
क्या कह रहे ਤੁਸੀਂ? ड्रेन नहीं ਹੁੰਦਾ, ਚਾਰਜਰ ਲਾ ਕੇ ਉੱਥੇ ਲਾ ਦਿਓ, ਪਲੱਗ ਲਿਆਓ 2 ਮਿੰਟ ਚਾਰਜਰ ਉੱਥੇ ਲੱਗ ਜਾਣਾ।
जाएंगे नहीं लोग?
ਕੁਛ ਨਹੀਂ ਇਹਦੇ ਨੀਚੇ ਤੋਂ ਕੱਢ ਦੇਣਾ ਤਾਰ, ਲਿਆਓ 2 ਹੋਰ ਚਾਰਜਰ। ਚਾਰਜਰ ਕਿੱਥੇ ਆ? ਪਤਾ ਸਵੇਰੇ ਇੰਨਾ ਕੰਮ ਕਰਿਆ ਨਾਲ।
ये हमारा चार्जर हो गया है शायद, है ना? हम दोनों का चार्जर स्विच हो गया। ये नहीं है ना तुम्हारा? मेरा एक बैटरी का
ਪਲੱਗ ਇੱਧਰ ਨਹੀਂ ਕੋਈ
नहीं है।
ਪੜੇ ਤੁਸੀਂ ਪੁੱਠੇ ਹੀ ਆ ਨਾਲੇ।
बढ़िया है।
बोर्ड भी रहेगा फट थोड़े ला दो साइड पर, ऐसे ला दो, ठीक है। देखो तो चल गया
हाँ चल गया।
है ना, बस ठीक है। बाकी थोड़ी सारे काम
स्लीप से हटा देती हूँ मैं इसे, स्लीप से हटा देती हूँ। सर का फील एप्लीकेशन है नहीं अंदर, और ये कहाँ से होती है, इसकी डिस्प्ले कहाँ से होती है, लॉक स्क्रीन टाइम, ये रहा, मिल गया मिल गया मिल गया। स्लीप वाला टाइमर कहाँ से ऑन करते हैं?
वो उसका बिल्कुल यहाँ देख लो डायरेक्ट इसको कट कर दो, यहाँ लिखो तेरा पावर स्लीप एंड बैटरी सेटिंग।
कंट्रोल पैनल में जाओ सर।
नहीं हो जाएगा। तो आपने 20 मिनट रखा हुआ है अभी, कितने मिनट रखना है? हाँ अच्छा,
बीच में ऑफ हो जाता है ना, ऐसा होता है।
लो अब नहीं होगा। होगा जो आपका लैपटॉप सही है, नहीं अभी होगा।
लेक्चर हो गया।
कोई दिक्कत नहीं, कोई दिक्कत नहीं।
Welcome back, everyone. Now we begin our first session of the day. It is our privilege to have amongst us today our esteemed guest of honor, Professor Sanjay V. Malhotra, PhD, FRSC, Department of Cell Development and Cancer Biology, and Sheila Edwards Linhart Endowed Chair in Cancer Research, Director of the Center of Experimental Therapeutics and Director of the Center of Radiopharmaceutical Research at the Oregon Health and Science University. Before joining OHSU, he was on the faculty of Stanford University School of Medicine and previously served as Director Chemical Diversity Division of the National Cancer Institute Experimental Therapeutic Program. He is a founding member of the Chemical Biology Consortium, a national drug discovery development program of the NCI-NIH, and served on the Joint Commission on Science and Technology of the Office of Science and Technology of the President. Dr. Malhotra's lab studies the science of therapeutics with the main focus on developing chemical tools to probe disease biology and discover small molecules that modulate the targets and provide pharmacological intervention. Dr. Malhotra obtained a PhD in chemistry and trained under Nobel laureate Professor Herbert C. Brown at Purdue University. His work has led to preclinical and clinical advancement of drug candidates. He has edited 5 books, is inventor on 20 patent applications, and has authored more than 170 articles. Dr. Malhotra is a Fulbright specialist and fellow of Royal Society of Chemistry. Ladies and gentlemen, please join me in welcoming Professor Malhotra to deliver our keynote address, What is Experimental Therapeutics? Drug Discovery History and Background.
Good morning, everyone. So as you heard, I am a chemist, chemical biologist, disease biologist, drug developer, experimental therapeutics, but I train as a chemist. So what I'm going to share with you is the research that I do, but most importantly, as the topic of this course, experimental therapeutics. Now, if you think of experimental therapeutics, like the same question, I'm standing here. So to make it easier for them, I will intermix English, Hindi, and once in a while Punjabi also. I will try. So उसकी कोई ज्ञान की requirement तो नहीं है कोई? Sure. क्योंकि online कौन है? I can continue in English. I have no problem. जो online वाले वो तो नहीं कहेंगे कि जी आप ये पंजाबी में क्यों बोलता पे सानू? Okay. I just want your permission before I start. All right. So yes. So we are together for whatever 4 or 5 days जितना Dr. Singh हमें allow करते हैं. And I'm going to share with you. It's recorded, so I'll walk and talk. So if you look at the title, since intentionally the title I gave is Advances in Experimental Therapeutics. So therapeutics by nature. Oh, I can wear that if. No, I'll stand here while you bring it. I'll stand here while you bring it. So as you see on the screen, इंसान है, दर्द होता है, शरीर है, पेन होती है. तो there are, ये philosophy नहीं है, reality है. So what is experimental therapeutics? Can you hear me now? Okay, this is better. I can walk and talk. So what is experimental therapeutics? Therapies and experiments, because therapy is ever evolving. If you look at the medicine, the field of medicine, the field of therapy as it has evolved. अभी भी है, पत्ते बांधते हैं जहाँ ज़रूरत होती है, pills लेते हैं ज़रूरत होती है, IV होता है जहाँ ज़रूरत होती है, but it evolved over the time, because as a chemist, and I think there was initially Dr. Verma was saying about, okay, you pick up, you learn, and then you contribute. And I'm just will make slightly a small addition to what he said, कि सारे नहीं, एक दो जो हैं वो ऐसे निकले जिनके नाम पे हो. My question is, सारे क्यों नहीं? Everybody should be able to do it, but we should try. So experimental therapeutics is basically therapy where it stands today and what all it takes to get to this stage. अगर आप देखें, अगर एक पिल भी हम लेते हैं सिर्फ टाइलनॉल, well, टाइलनॉल is not common here, एनासीन लेते हैं. What it took to make it an एनासीन, कहाँ से वो आई, क्या क्या उसके पीछे साइंटिफिक डिसिप्लिन्स इन्वॉल्व हैं, क्या क्या एक्सपेरिमेंट्स इन्वॉल्व है, किस तरह से होता है. ये तो मैं सिर्फ एक सिर दर्द की दवाई की बात कर रहा हूँ. But think of all the, and especially if you look at India and many other countries, but if you look at India, NCDs, non-communicable diseases, cancer, neurodegeneration, cardiovascular, it's on the rise world over, but especially in certain pockets of the world, it's more so than the others. So therapy निकालना इतना आसान नहीं है. First you have to understand therapy किस चीज़ की निकाले है ना, कि आपको पहले पता होना चाहिए कि मर्ज क्या है, तभी तो दवा बनेगी. And मर्ज क्या है, ये human body जो है, right? So first of all, to understanding what the human body is, how it functions itself is a huge challenge. We are, even though we may assume that we know a lot, itna advanced, but okay, we are barely scratching the surface. Nature is absolutely great. We do not even understand what nature and how it functions. So more we understand, understand, it makes it easier. It should make it easier for us to solve the problem. And problem in this case is whatever the diseases are, right? So all the various techniques, as they have been evolving, how they contribute to our understanding of the disease and developing therapeutics. That's basically experimental therapeutics. So during the course of this, all the lectures, probably I will try to cover, and Dr. Singh, Dr. Bala will try to expose you to various things. Especially my interest is to show you various different methods, techniques, technologies, advances that have. It's obviously each one of these is like a huge encyclopedic information, so it's not possible to cover in a week. But still, I will try to share information that will be helpful in trying to understand कि अगर therapeutics drug develop हो रहे हैं, तो what all it takes, क्या techniques involved हैं, at least so the latest what is happening around the world. So basically, if you look at the screen that I have. On the screen, what it shows is in the human body. The screen, what it shows is the human body. But what is human body? We have to understand what the pain is. When I say pain, which means क्या the disease क्या है, what the disease is. In order to understand, we have to understand what the human body is. Single brick of the human body, you're considering as a single DNA. It's a brick, और DNA से पहले जो है proteins हैं, RNAs हैं, right? वो सब हैं, वो क्या हैं? How that makes, and how do we translate the information that we have into a useful process that will tell us how it might be affecting the body. And that process then takes to creating something, merge की दवा, therapeutics, दवाई बनाने के लिए. So how to create that and what all it takes to create that. क्या validation होती चाहिए, किस तरह से होनी चाहिए before it goes back to take care of the health, human body. So this whole circle, this is a very simplified version of that, but we have to understand. So what I will be trying to share with you during the next few days is various different techniques which are involved here. So experimental therapeutics, this is a simplified definition if you look at, is the development of treatment strategies that effectively and very specifically help in treating the human diseases. The research involved basically it integrates various disciplines, as I was saying, and understanding, and each discipline is evolving. अगर हम देखें chemistry, from where we were to now, same way biology, pharmacology, toxicology, and not this is just the science, but then there are many other things which are involved, which I will touch upon when in one of my lectures later. And then as we have learned over the period, not one shoes fit all. The personalized medicine, because हर एक का makeup एक सा नहीं है, जो disease है उसका कारण एक सा नहीं है, because there are many proteins which contribute to a disease, and the cause of the disease in one person may not be the same as the cause of disease for the all other people. So as we understand more, that has become what is called the personalized medicine. We understand what is the as we understand the cause, then you can specifically design medicine for that cause. What is the personalized medicine? So basically this is, it focuses on the study and development of new treatments. Research in this is again, it's all understanding revolving around the human body. Now, how many of you have studied biology? Because I'll be covering quite a bit. Okay. How many of you have studied chemistry? How many have you not studied chemistry? Okay. How many have studied chemistry with molecules? Okay. The reason I'm saying is then I will try to use my focus accordingly, because if I start talking more into biology, तो ये क्या हो रहा है यहाँ पे? Dr. Singh कहाँ फंसा दिया हमें? So I will try to. You'll hear me joking all the time, okay? So it's कुछ serious भी हो सकता है, मैं questions भी पूछता हूँ. वो घबरा गए पहले. So anyway, so that's the reason I'm asked. So I will be covering chemistry and biology. So that's basically the idea, is to expose how we understand what we understand about the diseases, how we approach into these things, and how we develop that, use that understanding in various disciplines to develop therapeutics. Okay, that is the broadly speaking. I told you I'll ask you a question. Anyone, what is the drug? It's not a trick question. ऐसा कौन है जिसने अभी सिरदर्द की दवाई भी नहीं ली आज तक? मैं नीचे कर दूं क्योंकि मैंने ली है, मैंने अभी ली थी. How many of you have not taken any medicine? The lucky ones. So what is the drug? कोई बात नहीं, we have enough time. ये एक हफ्ता है, सोच के बताओ. कोई भी सब्सटेंस जो हम इनटेक करते हैं उसको ड्रग कह सकते हैं. पॉइजन किसको कहेंगे? Good. Anybody else? Yes? Close. Very good. Close. It's a very simple question. Yes, but what is the medicine? I'm sorry, say it again. Very true. But what is the drug? वो ड्रग किसको कहेंगे आप? Very close. How does this sound? Right? So जब मैंने पॉइजन का इसलिए कहा था कि पॉइजन भी रिएक्ट करता है. Right? Drug should have biological response, favorable biological response, right? जो poison है, जो toxicity जो है बहुत टाइम पहले I think a couple of centuries ago, किसने कहा था toxicity is in the dose. अगर आप फूड भी जरूर से ज़्यादा खाएं, it becomes toxic, it becomes a poison for you. जितना पचा सकते हैं, जो अपना कर सकते हैं, है ना. So basically, any compound or any material that you take in, it has biological response. Then it's a drug. भूख लगी है, not philosophically, सच में आप देखिएगा अगर आपको भूख लगी है, आपको खाना नहीं मिल रहा, आंतड़ें अंदर से खींचनी शुरू हो जाएंगी, right? That's a pain, right? आपने खाना खाया, थोड़ा आसान आया, तो that's a drug for you, right? आपने पेन किलर लिया, वो पेन को रिलीव किया, तो उसने बायोलॉजिकली कुछ किया, it did some biological thing inside the body, so it's a drug for you. But if you excess of food, the reason I mention food is because if you take excess of food, बदहजमी हो जाएगी, right? Then becomes a poison. So drug is basically any compound. Here for simplified version I say compound. Anything that you take in that has a favorable biological response is a drug, right? And over the years, if you, yeah, please go ahead. I'm sorry, say it again. हाँ, बायोलॉजिकल रिस्पॉन्स है क्योंकि आपको उसके बायोलॉजिकल मॉलिक्यूल्स क्या हैं, केमिकल रिस्पॉन्स हो रहा है. योर वंडरफुल क्वेश्चन, एब्सोल्युटली. सो केमिकल रिस्पॉन्स कहीं या बायोलॉजिकल रिस्पॉन्स, बट वी आर टॉकिंग अबाउट ह्यूमन बायोलॉजी, इसलिए मैं कह रहा हूँ इट गिव्स ए रिस्पॉन्स टू दैट. इट कुड बी ए केमिकल रिस्पॉन्स, इट कुड बी बायोलॉजिकल. Just to differentiate chemistry, when we focus small molecules. When we say biochemistry, we focus on large molecules, RNAs and proteins and DNA and antibody. So you're right. A response that we get, biological response, because we are talking about human biology. That's right. But theoretically, you're right. It's a response that we get is a response to the body. When I started, why I got into chemistry? When I started, I had a wonderful teacher, and यहाँ lecture hall में नहीं है. When I went to जहाँ मैं था, वहाँ पे every room had a periodic table, like we, right? And when my professor walked in one day, he said, "This is life." And now अभी आप थोड़ा सीखना शुरू हुए हैं. How is this life? Right? Periodic table है, उसमें तो molecules के अंदर सबके symbols लगे हुए हैं. How is this life? But think for a second. Any physical entity that you can think of, जिस चेयर पे आप बैठे हैं, इस बिल्डिंग पे, आप पत्तों को देखें, एनिमल्स को देखें, इन एनिमल्स को देखें, anything that you can touch and think of has come out by combinations of those. आप प्रोटीन्स देखें, 22 प्रोटीन्स जो नेचुरली बनते हैं, कार्बन, हाइड्रोजन, नाइट्रोजन, सल्फर, ऑक्सीजन, राइट? प्रोटीन्स देखें, आप डीएनए देखें, इट इज़ अ कॉम्बिनेशन ऑफ दैट. आप मेटालिक रिएक्शंस, मेटल के मेटल प्रोटीन्स हैं, जो भी आप देखना चाहें, एनी फिजिकल क्रिएशन यू कैन थिंक ऑफ, उसका जो मेकअप है is by combinations of those things. There is nothing outside of those periodic, as best as we know. Then I said, well, no, he's right, that's life. If you can understand the functions of the periodic tables में जो सारे हैं एकदम अच्छी तरह से, you can go in any direction you want. What is biologist? As I was telling him, what is a biologist? DNA. And as a chemist will say, ठीक है, no, it's a combination of सारे जो atoms हैं उनका, वो अपने एंगल से देखे. तो so basically so that was prompted me कि chemistry is fun because if you can learn more and more you can learn, क्योंकि आप एटम से मॉलिक्यूल्स बनाएंगे, मॉलिक्यूल से लार्जर मॉलिक्यूल बनाएंगे, फिर you can go to environmental, you can go to nano, you can go to human body. It's basically that. So that is what I was taught actually, and that stayed with me. Actually that is chemistry. It's more fun to be creative than to be I mean, all fields are good, but that attracted me. So yes, chemical reaction जो आप कह रहे हैं, true, chemical reaction or physical reaction, but response है. It is a response to something that you take in, which is favorable to your disease, then it becomes basically a drug. So historically, जहाँ मर्जी हैं, if you look at various different cultures, जहाँ से भी हैं, where we are here, for example, कुछ नहीं होता था, क्या था कि हल्दी पिला दो, हर चीज़ की दवा हल्दी है, नीम के पत्तों को बांध दो, सबकी दवा रहने करती. So it was more of observational. People across the world, different cultures, when that's how they start basically observing the nature. So I'll just tell you basically how this experimental therapeutics and this whole drug discovery thing evolved. So it was basically observation of के यहाँ पे जिस तरह से हम लोग कहते हैं कि नीम और हल्दी हमारे this part of the world is common, and in the Western world it's called grandma's chicken soup. Anything happens, just give the chicken soup, you'll be fine. So that was their way of, but basically as observation, it was all observational. क्योंकि जो चलते आ रहे हैं उन्हें ऑब्जर्व किया कि ये पत्ते बांधने से ये ठीक हो जाएगा, ये हल्दी का इतना लेप लगाने से ये ठीक हो जाएगा, बट बेसिकली those things, whatever those were observed, those things were used, they had therapeutic applications, right? So the industry, the observations evolved that way. जो मेडिसिन की अगर हम हिस्ट्री देखें, these were folk medicines. आज की तारीख में भी you go close to the Himalayas या the African Amazon में, जहाँ भी not the urban area से जहाँ भी दूर जाएं, there are people, they don't go to a modern therapist. उनको पता है कि कुछ शरीर में कुछ दर्द है, कुछ कहाँ करना, उनको मालूम है कौन से पत्ते का कौन सा स्टेम लेना, किसी लीव प्लांट का, या कौन सा पत्ता बांधना, खा लो पी लो, कितना उबालो, क्या मिलाओ करो, ठीक हो जाता है. So these were all folklore medicine जो इवॉल्व हुई, ऑब्जर्वेशन करनी शुरू हुई. And based on, so this was a couple of centuries ago. That's how it evolved. It started to evolve, and more then other parts of the science, for example chemistry, started to become more and more advanced at that time. People could not only just take this natural product soup, but learned that within natural product, अगर आप soup भी लेते हैं, इसमें not just one molecule, it's a mixture, it's a combination of many molecules that you use. Then it becomes easier to identify those molecules. The techniques were developed to identify those molecules. Those molecules started to be extracted from those plants. उनका analytical chemistry के लिए tools आसान हो गए बनने के लिए. आपने निकाल तो लिया, मगर identify कैसे करेंगे कि what is the structure of the molecule? Those techniques, or to identify that, you have to have the techniques to know how to identify. So in parallel, all these sciences, all these know-how was being evolved. So 19th century में आप देखें, late 18th century से 19th century में, this is how the development happened. And then slowly started to form an industry for focused scientific areas. जो हम कहते हैं आजकल pharmaceutical industry started to evolve in 19th and 20th. The companies like Bayer, जो German companies और British companies, उन्होंने इस तरह की अपने टेक्निक्स से उसको इम्प्लीमेंट करना शुरू कर दिया कि अगर प्लांट हैं तो प्लांट में से एक्सट्रैक्ट्स को निकालो, उन एक्सट्रैक्ट्स को अगर आप निकालेंगे, आइडेंटिफाई करो, सो यू कैन से कि विच मॉलिक्यूल इज एक्चुअली द ड्रग मॉलिक्यूल, रेस्ट इज जस्ट द combination which is needed to make a formulation. So that became more and more advanced at that time. As that became, and then again started to convert into some form of application. So if you look at, for example, I have given here जो ephedrine है, this was Park and Davis, a British company, a long time ago. This application, cardiac arrest, अब है उसकी application, which is more recent. But that was the molecule which was used for sepsis, skin where भी है, and that was again based on observation. कि जिस भी plant से लिया करा, and then when you put it on there and you see there is a reaction, favorable reaction, and say okay, this is actually a medicine for that favorable reaction, sepsis के लिए. And then more and more studies came, and now it's used for for cardiac arrest से पहले it is used for अपने डेंटिस्ट के पास में गए हैं तो आपको एनेस्थीसिया देते हैं, राइट, उसको नम करने के लिए. इट हैज़ दैट एप्लीकेशन. सो इट उसकी एप्लीकेशंस जो हैं, सेम चीज़ जो एक्सट्रैक्ट बनी उस पर रिसर्च होना शुरू हुआ. उसकी एप्लीकेशंस बनी निकलने लगी कि वेयर ऑल द प्लेसेस दैट कुड बी यूज्ड. एंड सी आई हैव जस्ट शोन वेयर इट इज़ नाउ. इट इज़ जो है वासो कंस्ट्रक्टर. इफ यू गो टू डेंटिस्ट, वो आपको देते हैं, नम करते हैं बिफोर दे स्टार्ट देयर विद देयर टूल्स प्लेइंग विद योर टीथ्स. सो जो नम करने के लिए है, दिस इज वन ऑफ द थिंग which is you. But point being, this was all observational. It helps the sepsis, ठीक है. And then if you can extract the molecule, then you start studying its application in different areas. कि कहाँ उसके beneficial. And this was mostly on observation. Barbituric acid, barbiturates जो हैं ट्री प्लांट्स में जो एक्सट्रैक्ट होते हैं, अगेन 19th centuries में जो है, you know, a lot of these things started to become more and more. These were observational, going to the industries, taken over by the industry, and then industry trying to find their biological activities, biological application. If you see here, for example, barbituric acid, that and I don't know if you have heard the name of Emil Fischer, who diethyl barbituric acid, barbarita, जो उन्होंने 1904 के अंदर उन्होंने उसकी एप्लीकेशन दिखाई थी. But that then this is how where we are. It's now a sedative. Sedative, depending on उसको convert कर दिया गया sedative में. It can be used as a sedative. So observation, it started with simple observations, and then that led to more proper applications. Look at, I'm sharing with you a few examples. Morphine जो है, you know, for again extracts from plants, right? If you go to jungle, or quote unquote jungle, but if you go to the remote areas, people there know कौन सी लीफ के पत्तों को लें तो आप अच्छे से नींद सोएंगे, अच्छा नशा चढ़ेगा, right? So उसमें क्या है, what is in those leaves? They know that, but they didn't know the extract. And the modern science, quote unquote modern at that time, started to extract those molecules, tried to extract them you know, from various different parts of the plant. आप देख सकते हैं, आप स्टेम से लेके लीव्स, जो उसके सीड्स हैं, from all of these more and more research started to develop, and those then converted into more. Now today it's an alkaloid, basically opioid alkaloids which are in there, which are used there. So alkaloids जो है, morphine जो है, now obviously these are observational recreational drugs, dam maro dam, but then that started to become more into the medicine. कि उसका जो observation थी कि it obviously, अगर हम वो भी लेते हैं, अब बात करें, I just lightly gave you dam maro dam, but basically what is happening, you take that, it goes inside, it is doing biological reaction. उसका डोज इतना ज़्यादा है कि it numbs down जो proteins और जो signaling pathways हैं, उसको numb down कर देता है, and people feel high, right? But उसी को convert करके, उसका right dose निकाल के, उसकी right application निकाल के, it becomes a medicine now. So this is the morphine is used now for managing the pain, because for in cancer patients, for example, because those drugs are highly toxic, बहुत painful होते हैं, because, and I'll come to that, okay, those, because you know, it's a chemotherapy, for example, is we are bombarding the body, the cell with a toxic chemical, तो उसके बाकी जो natural, जो normal cells हैं, normal part of the body, उसको भी काफी इफेक्ट करता है. There is pain. To manage the pain, morphine is actually prescribed now, but इसकी observation started a couple of centuries ago. So the point I'm trying to make is, as the organic chemistry, extraction chemistry came out, उसके techniques बननी शुरू हुई, तो जो molecules थे, वही जो सिर्फ हम सूप ही पीते थे, उसमें से single molecules started to come out. उन्हीं molecules में से उनकी biological applications की study होनी शुरू हुई. It started to evolve धीरे धीरे करके कि क्या है, and these are more examples. Now the reason I'm sharing the example, पहले जो मैंने आपको दिखाया, that was because the analytical techniques become so available that you can separate the molecules, and you can identify कि जो pharmaceutical या medicinal therapeutic application है वो कौन से molecule की है, because you had those those tools available. Same way, extraction solvent technology became more and more available people because that part of the science started to develop. And then now if you take pura as a soup peene ke bajaye, if you can extract a molecule, if you can crystallize a molecule, you can give applications differently. The crystal chemistry, the extraction for example, was known in the 18th century. 1747 में जो है, they knew कि extract करके crystal को कैसे बना सकते हैं. How you freeze it and convert something into crystal, right? Crystal की applications we know, sugar खाते हैं. That's a crystallography. जो crystal उसको मतलब crystallography in the sense to converting into a crystal, जो solvent है उसको लेके. So that technique became more and more. Sucrose जो लिखा है, no, I wrote the crystalline sucrose वो was shown in 1747. How you can take the sugar and it becomes actually a crystal, right? So that when that technique became more and more, solvent extraction became more and more, knowledge came out, and now the same. Now think of कि पहले जो plant में से सिर्फ extract करके पत्ते ही खाए जाते थे, उनका soup ही बनाया जाता था. अब उसमें से you can actually extract different molecules, and then you can study the application of different molecules. Now I'm so far touching only the chemistry side and the analytical chemistry side. I'll come to the biology side. Study करने के लिए, what are you studying? You need to know what you're studying, right? I didn't do anything. So as these technologies, these knowledges this knowledge became more and more available in different disciplines, different areas, applications started to become. Don't worry, I'm going slow. I'll jump quick, bring you to the currents. But I'm just trying to give you an appreciation of how it takes time for science to move. And then you see now opioid, many parts of the world, opioid is a huge problem. Problem is because regulations के कारण, because it gives high and people are using it left and right, and without knowing उसके जो negative consequences, and that is why it is a problem. But it also has biological application. That's why it's a problem. Now can that application be converted for a useful purposes, and then it becomes a medicine. So opioid based medicines, again morphine, right? जो civil war के दौरान, it was one of the things which was more used, but then slowly regimented तरीके से it developed and became into a medicine. I'll give you a few more examples and then I'll bring back quickly to what is the current जो modern chemotherapy, what it is and where we are, and how that works. So quinine, malaria, this part of the world we know the term malaria very well, western part eradicated a long time ago, not. So malaria के लिए cinchona bark जो है, quinine जो है, quinine is basically a molecule which is extracted from the bark of the cinchona tree, right? And that was observational, because if you take that was again purely observational, that if your cinchona bark जो tree है उसके bark जो है, if you किसी को malaria हुआ होगा, these are all human observations. किसी ने गलती से, क्योंकि farming is the main thing, वहाँ उठा के खा लिया कर लिया होगा, उसका बुखार ठीक हो गया होगा, there is something in there. So that observation led to actually identifying what is in their bark. That led to cinchona, right? That led to quinine. Now these were based initially only on the extraction, because extraction technology was available, extract करके बना सकते हैं. But the fact is, to spread it more places, then organic chemistry, analytical chemistry became more and more accessible and more advanced at that time. कि उस cinchona bark, because bark is not one molecule, उसमें hundreds of molecules कुदरत ने डाले हैं. But how come you just get to quinine? So you extract it, analytical chemistry advanced होने लगेगी, you extract various techniques and you identify quinine is the molecule, which is the one which has the pharmaceutical application. And that is how that evolved. And then as you can see, the cinchona bark in the 17th century, those observations were made in 17th century. So think for a second, I mean I'll bring you back to, but again for the appreciation, think for a second, okay, we are in 2025 as per the modern calendar, and of course world existed even before that. Up until 17th century, कुछ ज़्यादा पता नहीं था. There wasn't much information known. So this is all past 2.5 to 3 centuries. और उसमें भी अगर देखें, I'll share with you examples of इस past 3-4 decades जो advancement हुई है. So on the human scale, to make an advancement, it takes a long time. Long time. So as you can see here, कि ये करते कराते यहाँ तक पहुँचते हुए भी ये सिर्फ 17th 18th century का है. Give you an example of 1904. तो ये सोच के देखो कि 1904 पहले 1904 years, if you start with that end, prior to that there was no such information available. लोग क्या करते होंगे? Therefore the lifespan was also in 20s and 30s. Lifespan wasn't that much at during those times because the disease, I mean, if you look at the history, if you read the history, famine, बुखार आया, जो कुछ भी हुआ उसके कारण इतनी diseases फैली, and then and the population got wiped because there was nothing to fight against. So our understanding of what we call the modern science, as you can see, is very recent. It's evolving and it's very recent. While this machinery is very complex, and so therefore, again, I'm connecting you back to just to give the appreciation of experimental therapeutics. Okay, techniques that has brought us to where we are today has taken a lot of hard work by the human as a race to bring us to where we are right now. So these are just just to see. And then the first synthesis. Now quinine extraction was known in 1747. Think for a second how the speed with which it moves. 1747 it was known that cinchona bar has a therapeutic effect, right? But the molecule to synthesize first synthesis was done in 1944. 200 years to make a molecule. So just imagine on the human scale, our understanding, our knowledge of each field, it takes a lot of, and 200 years to come up with the synthesis of that molecules. Uski application was known 2 centuries ago, nearly 2 centuries ago. So that's how, just to give you a few examples, the appreciation of the, what has taken, the historical background, what has taken, and how the science have evolved. Now again, this is, because many, as we call fields, most of you are chemists, all of you are mostly chemists in this, so we are talking about chemistry focused. But the fields, when we talk about, as I mentioned, biology, even biology you can subsect depending on किस तरह की biology आप कर रहे हैं, pharmacology, toxicology, cancer biology, chemical biology, in general I call it disease biology. Each one can be broken down. Immunology now, which is the field which is evolving so rapidly. All of these are highly complex and were not advanced. So this is just based on giving you appreciation for the chemistry. So these are all observational again. And then again, as human races, झगड़े होते हैं, wars होती हैं, तो दुश्मन के खिलाफ many things are used. So I'm showing you here example of one of the observations that was made during the war. So 1917, that's over a century ago, 108 years ago, right? Then troops in Belgium, they were fighting, they observed that there was some smoke coming out, and that with that smoke they had blisters all over their body कुछ घंटों बाद में, and pain, excruciating pain, and many of those people died, troops died. Then the question was, what was that smoke? Anyway, a lot of science, observation, analysis happened. It turned out that it was basically mustard gas which was used against them. And mustard gas, as we talk about chemical weapons, usme bhi mustard gas is used. So basically what it does is it attacks your body ke cellular functions and has all this toxic effect. So that observation was made in 1917 after that attack, that they see that the gas has something happening, which is causing them for all the blisters, their body not reacting, reacting to that gas and having very toxic effect. So the first use that happened, and there were 10,000 people who died because of that smoke. And that smoke basically was nothing, that mustard gas. Now when that happened, then obvious curiosity is, how does it react to the body? What does it do to the body that it caused such a severe reaction and 10,000 people died? So there was obviously curiosity, scientists, academics, curiosity from them to see why it is that smoke was so toxic. So two scientists at the Yale University, World War Louis Goodman and Alfred Gilman, they were medical doctors, medical researchers. They were curious to see that what was that happened. And when that happened, what was the reaction? Body reaction to that mustard gas. And they were curious to see how do we measure the effect that happened. So what they, again, a regulatory, you have to go to the government, you have to get the approval to get the samples of the troops, those who survived and those who are affected. So it took them a couple of decades or more to get those samples or to get the information about the other troops, those who are affected, and study that information. So what they found out was that, as I have noted here, that the number of immune cells in their blood was affected. It was affecting surprisingly low, the white blood cells. And so when they started to think, if that happened, then maybe there could be a useful therapeutic application that can be developed with the mustard gas. And with that, they started doing experiments. That if that happens, then probably it could be used for some biological disease that we don't know of. And then they started to treat. They did many of the the lab studies, and they did the animal trials. They found the human volunteers after the successful animal trials to see that if they see some positive effect on the disease, and specifically they were studying leukemia and lymphoma, the cancers of the blood cancers. That's what they were researching on. And so based on the medical records that they observed, based on the observation that the immune cells specifically number goes down by the treatment, and since they were studying leukemia and lymphoma, they started to develop method to study by treating leukemia and lymphoma cells and saw the positive effect. They took that knowledge to start doing animal studies, study the same effect in animals affected by leukemia or lymphoma, and see that again they observed very positive outcome by the treatment. Then they found some human volunteers who voluntarily, they had the disease and it was not affecting, and then say, okay, fine, there is nothing else left, try this. And those human volunteers also. So the first time, the formal record of giving a known, well-studied, at that time well-studied, a candidate to a patient with cancer was, as you see on my screen, it was at 10 a.m. on 27th of August 1942 to protect the identity of the patient. It was called John Doe, no name given to that, was treated with that mustard gas. And that cancer patient started to improve. started to feel better, started to eat better. Okay, his condition started to improve. And unfortunately, he didn't survive after some time. But the reaction was that by using that mustard gas, by using the chemical that they converted into a drug, there was positive response by the cancer patient. That is the first known record of chemotherapy that you hear so frequently now. So just to give a historical background, so centuries and centuries, nothing known. 17th, 18th, 19th century, some observations. That observation leads to systematically developing some approaches, some strategy, and that is what led to the first known chemo treatment. So 1925, so that's what, 83 years ago, was the first chemotherapy was given. So think about human race, we are in 2025, it's only very recent. Actual chemotherapy jo hai, and even though that we know that there is a lot more that needs to be done than has to be done in order to cure many of the diseases that we have. So anyway, because the analytical chemistry was known, known in the sense there were tools of analytical chemistry available to be able to identify the structure of a molecule. This is, as you look at the structure of mustard gas, it's a very simple molecule, but see how much effect it has on the human body. Blisters and such a toxic effect, but it's a very small molecule. So once that was known, so I have shared here a few more examples of similar molecules that were developed. So when that was there, people started obviously knowing that if this molecule have such a huge reaction, positive or negative, depending on the dose and how you treat and what you treat, then there is potential that similar molecules can be converted into drugs, therapeutic application, and of course, creative chemistry mind started to come up with similar structure. And if you go into literature, there are, and you'll hear from Dr. Singh and Dr. Bhalla about the metal-based drugs that are being used and for various treatment. But this is how it evolved. Simple observation. And then again, I have seen that chlorambucil, one example, which was from, Dr. Haddow, had shown that its application from UK. But see, cisplatin is, platinum-based. This is one of the structures, and then you can make various variations, like carboplatin. In 1972, it was patented. But again, as you see, the speed with which now people thinking of more molecules that could have biological application speed up, because people know how to make molecules, people know how to analyze molecules. Then there was started to develop testing methods for testing those molecules. So it became faster and faster to identify more molecules. And I've just represented a few examples. But if you go into literature, you'll find hundreds and hundreds of these molecules which have been around just simple mustard gas, have been designed and tested and for various different diseases. So just to give you an appreciation of how just observational, and still it's observation. Observation has to be, as a scientist, you have to be very observant. But as simply an observation-based medicine to now systematically developing medicine that could be. Now this is all the chemistry side of it. That you have chemicals which are again coming from natural resources. आप क्या खाते हैं, क्या पीते हैं, उसका reaction to the body. Based on that, it became formulation. कि कितना खाना, क्या मिलाना. You make those formulations. And then as the analytical chemistry side, as the organic chemistry side, as the techniques of synthesizing molecules, as the techniques of analyzing molecules became more and more prevalent, more and more easier, it started to speed up the process, to be looking for now not just few molecules which are coming from natural products, but you can now simply design different molecules. You can synthesize different molecules and test for various applications. And that application, again, in this case, we are talking about drugs for the human diseases. But again, that's how, treating even the plants. जो है organic manure जो है, It is what? Manure, simple manure की जगह पर organic आप दे रहे हैं. जो आपके कीट पतंगे जो हैं, you know, those even for the plants, if the plants के कारण जो पत्ते खा जाते हैं हो जाते हैं क्या है? It's a disease for those, right? You started, but point being, as a chemist started to develop, thinking of how many different molecules that can be synthesized, they can be tested, and their useful application. Now, when we talk about drugs, we are talking about drugs, about human drugs, and that's what we'll be covering. But that's again, it's a drugs for the plants, for the animal. They are drugs. If you can, you give the pesticides, you spray them, and they are healthier. Those are actually drugs for those. So, as the field of chemistry, I'm focusing on chemistry, and probably what we could do is, if you want, take 5 minutes break, and then I'll cover the chemistry, the biology side of it. So, but before you go, if there is any question, but that's just to give an appreciation of how things evolve, so that we understand कि अगर drugs हैं तो क्या है, where they come from, and how many different disciplines have to be brought together in order to understand the disease, the causes of the disease, and then to develop something which is safer and effective. Okay, any question? Anything you want to ask? Anything you want to share? Yes, go ahead. I'm sorry? हैं, but unfortunately कम है reported, क्योंकि जो उसमें investment, focus, determination, dedication is needed. It, for whatever reason, came from various parts of the world, and many of those were outside of India. Most of those, unfortunately, were outside of India. It's not that, I mean, if we see, the thing is, if you look at the history anywhere, if there is knowledge, I'll give you an example. That knowledge has to be protected. That knowledge has to be nurtured. That knowledge has to be brought together to the world level in a way that is effective and useful for humanity at large. And that, whatever emphasis उस पे दिया गया नहीं दिया गया, observations have happened, no doubt, but उसको किस स्पीड से आगे लेके जाना, उसको आगे लेके जाने के लिए आपको जो डेडिकेशन चाहिए कि कुछ भी हो जाए. अब many of these discoveries, for example penicillin, right, उसने अपने आप को सबसे पहले दे दी थी, but you need that guts के, you have to have those कि I'm absolutely right, this will work, I'm so right that वो कहता है I will be the first patient to take this. वो guts भी तो चाहिए. I mean वो it's not as simple to answer, but yes, you are correct. Most of these, as we see, the modern has come out of places, various parts of the world which is away from here. It's being recorded, otherwise I can give a non-political answer. It takes a lot. So as you see here, there are many reasons, as you see here. So that's a good question. Forget about, political, not political. The part is, it's a multidisciplinary process. What I'm talking right now, science, and even in science, I'm talking only the chemistry. You take 5 minutes break, I'll come to biology. And that's only science. Then we will come to the regulatory side. Then we'll come to the geopolitical side. What all it takes to get a drug to the market. And there are many, many. Insan is a complex animal to begin with, So anyway, I'll talk after that. So yeah, please. What else? Yeah, go ahead. किसने कहा आपसे कि they create problem? You have inside sources?
Not really, but there are some complications also that come with, let's say about
It's not that they intentionally created. These are the side effects. You see, these are the side effects of any medicine that is developed, because medicine, as you will see during, I'll talk about that, because the idea is, well, I'll come to that, but just think for a second. If a medicine is developed, it's developed based on what we know today. We as a scientific community, we as a humanity, as we know today that this is the cause. And we try to, with all sincerity, with all the resources, with all the knowledge we have, we try to combat that based on what we know to do that. That's our collective understanding. But do we really know exactly कि उसके अलावा वो कुछ नहीं करेगी? Think for a second. एक cell, I'll show you a picture I think in the next lecture or something. I'll show you a picture. Cell is a soup of thousands of proteins and many other things. If you develop a drug for a protein target, इसका मतलब ये नहीं कि ड्रग किसी और चीज को टच नहीं करेगा. That's why it's a very complex thing. You have to do a lot of toxicity studies, and the toxicity studies are to make sure that it is safe. ऐसा नहीं कि वो एक चीज का जिसको बचाएगा मैं दूसरे से मार डालेगा. So side effects are, the goal is always to minimize the side effect as best as possible so that they are not overburdened, that the positive effect is nullified or it's become less relevant. So that's, I believe, universally, every scientist working in this area is the interest to come up with something which has positive value, positive effect, but not have negative effect to the extent कि उसका कोई use ही ना करे उसको. That's not the goal. Yeah, go ahead. जी. ये बनाने वाले की रचना बहुत मजेदार है. You want an answer now? I can give you that, but I'll come to that. Keep that question in mind, and I'll explain to you how आपको मालूम है ह्यूमन बॉडी जो आप खट्टा खा लीजिए, आप मीठा खा लीजिए, आप कड़वा खा लीजिए, आप जितना मर्जी तीखा खा लीजिए. ये तो मैं बड़े सिंपल सी चीज़ें बता रहा हूँ. आपने खाना है, जो चने आपने चबाने हैं, फिगरेटिवली स्पीकिंग और रियली स्पीकिंग, जितना कठोर खाना है उसको भी चबा पाना, ये सब कैसे होता है? Keep that in mind when we come back. I'll explain to you. ये जो रचना है बहुत मज़ेदार है, बनाने वाले की रचना. So human body at different parts has different pH. जाते हैं वो उसको तोड़ फोड़ के कर देती है एकदम छोटे छोटे molecules में. Then it travels to different parts of the body to get to what gives you the energy. ATP ये सारे नाम सुने ना आपने, कि एनर्जी में कैसे कन्वर्ट होता है, वो सब कुछ जो है कैसे ब्रेक डाउन होता है. वो ब्रेक डाउन करने के लिए किस जगह पे क्या पीएच होना चाहिए, उतना ही पीएच है. ये मशीनरी बहुत मज़ेदार है, बनाने वाले की रचना. I'll come to that. So that is how it works. So आपने तो बड़ा सिंपल क्वेश्चन पूछा कि हमने एक फॉर्मुलेशन लिया, ठीक है. फॉर्मुलेशन तो हमने एक्सट्रैक्ट करके सिंगल मॉलिक्यूल ले लिया. अगर पत्ता लेंगे तो कैसे होगा? हमको तो कुछ भी नहीं आता, जो बनाने वाला है उसने तो सब कुछ बनाया हुआ है अंदर. दैट पत्ता ब्रेक्स डाउन, दोज मॉलिक्यूल्स गेट सेपरेटेड. द मॉलिक्यूल विच आर ऑफ लेस यूज दे गेट फिल्टर्ड आउट. The molecule which is of use goes and affects on the side where it should be affecting and giving you the positive or negative effect. So ये जितनी भी हम इंडस्ट्रीज देख रहे हैं ना, रियली स्पीकिंग, जितनी इंडस्ट्रीज हम देख रहे हैं ना आज के फार्मेसी, हम एक्सट्रैक्शन तो एक चीज़ है, सिंथेसिस, एक्सट्रैक्शन, बायोलॉजिकल रिएक्शन, राइट, फिल्ट्रेशन, जो फार्मेकोलॉजिकल, I'll come to, I have a whole lecture on that, I'll come to जो what is pharmacology. ये तो हमने नाम दिए हैं based on what we know. As we are learning, we are giving this name. Body does that, has been doing this for generations, और millennium से करती आ रही है. ये तो हम अब समझ रहे हैं कि वो कर क्या रही है, और जितना थोड़ा बहुत समझ में आ रहा है. So all these things happen. If you take the body, body goes. So let me tell you the quick answer. आपका जो गट है उसका pH क्या है, किसी को मालूम है? आपने सल्फ्यूरिक एसिड देखा है? देखा यानी सल्फ्यूरिक एसिड आपको क्या मालूम है? उसका pH क्या है? कॉन्सेंट्रेटेड सल्फ्यूरिक एसिड. ये आपके गट का pH है. जो आप खाते हैं ओरली, आप जो कुछ खाते हैं सीधा वहाँ जाता है, उसको तोड़ देता है सब कुछ. जैसे ऊपर जाते हैं उसका pH चेंज होने लगता है, तो फिर उसमें फिल्टर होकर निकलने शुरू होते हैं. देन इट गोज टू प्लाज्मा, इट गेट्स इनटू द ब्लड, इट रीचेस टू द साइट ऑफ दैट थिंग. सो हम जितनी भी इंडस्ट्रियों के नाम ले रहे हैं वो सारी अंदर बनी हुई हैं. सो दैट्स हाउ थिंग्स. सो इफ यू टेक लीव्स फॉर एग्जांपल, लीव क्या है? आप धनिया पुदीना लेके खाते हैं ना, लीव ही तो खा रहे हैं आप. व्हाट हैपेंस? उनमें भी थेराप्यूटिक वैल्यू है, तभी तो हमको अच्छा लग रहा है. अच्छा इज ए फीलिंग, बट वो एक्चुअली कुछ ना कुछ पॉजिटिव कर रहे हैं, तभी हम चल रहे हैं. समथिंग पॉजिटिव इज हैपनिंग. यू टेक द फूड, राइट? यू टेक सैलड फॉर एग्जाम्पल, यू आर ईटिंग लीव्स, तो उस लीफ में और इस लीफ में क्या, पत्ते तो अंदर जा ही रहे हैं. बट दैट्स हाउ द बॉडी इज मेड. इट्स, आई मीन, more and more you learn science, you just go, wow. I mean, it's such a beautiful machinery created. So all these industries are inside your body. All these functions are happening inside your body. Okay, if you want, just take a few minutes break. If you want to, grab pani pina hai, karna hai, otherwise I will continue. I have no issue. I can continue non-stop, but if you just, I thought probably ab subah se aapko logon ko band ke rakha hai andar, so you can just take a few minutes break, and I'm ready. My slide is ready. Just take a few minutes break, come back, and I'll start again. क्योंकि कैमरा है, मुझे ही फेस कर रहा है.
नहीं फेस है, बट जो लाइव में चलेगा वो नहीं चलेगा.
थैंक यू. मैं वो मुझे पता है, मैं कैमरा हटने
वो फेस
वो यही चलेगा. What do you guys do? You're in your PhD program?
Yeah, we are also scholars under Professor Singh.
Uh, Gurjas reason. So what do you do? What do you make? आप क्या करते हैं? क्या? What kind of project? What do you do?
Our work is on basically.
Triazole synthesis.
Yeah, but then we study the biological,
What kind?
biological study. Basically metal sensing, we do metal sensing.
Extraction. Metal extraction.
Sensing how that metal and the compound which we have actually created is acting as a ligand. And the sensing.
Okay, so I was telling you about the briefly, very briefly about the developments in the chemistry side. Okay, whether it's a synthetic chemistry, organic chemistry, natural product chemistry, various techniques जो भी हैं. A few I just briefly mentioned how it helped in trying to understand contributing to the therapeutic development or drug development. At the same time, obviously, those who are more interested in the biology started to study more and more of biology, and I'll tell you more in detail in few lectures after now. But so what is chemical biology? So I'm trying to stay focused on the experimental therapeutics way. So under application of chemistry and understanding of biology together, when you are trying to use chemical tools to understand the biological problems, it brings together, and in loosely it's a marriage of chemistry and biology, and slowly a new field in past, I think at least now more formally in last 30 years, which has evolved, is what is the chemical biology. And as more and more understanding of chemistry and biology became clearer and clearer, it evolved more and more, it led to advancement much faster. So if we look at in past few decades, in this case I have just a timeline of over a century now. If you look at this, how these understanding of our chemistry, mostly focus as I'm showing chemistry and biology understanding, has slowly contributed in developing new drugs. And this is basically, as you see, just a timeline, which again I've taken from the AACR sources, from the published information. But you see, simply when the animal models came, that you can study your molecules inside the model and can get some yes and no answers, whether this molecule, your molecule of interest, is working or not, what kind of thing it is doing. So as you see, it started to evolve more and more the transplantable so this is for keeping mostly cancer disease as a focus. Transplantable tumors, so you can take tumor and you can plant inside an animal, and that's how Gilman and Goodman were able to do their animal studies. But that knowledge initially started in 1912. It is possible. It was not necessarily done for cancer, but you can take actually a human tissue and you can put inside an animal and you can study the reaction there. So that knowledge started about a century ago. And then as you see, that has then nitrogen mustard gas, as I was telling you. And then you can see these are just a few examples of how things over the years have evolved in trying to use whatever knowledge modern at that time. Modern in 1900 is different, modern in 1925 is different in what we call modern in 1975, and different than what we call in 2000 and 2025. So in the given time, whatever knowledge was available, based on that, they tried to develop some therapeutic applications, and those became drug. Now, as I mentioned, as our understanding of this was evolving from the simply observational science to actually understanding inside the human body, became the more tools, more methods, more experiments, more strategies, more approaches evolved as great minds came and then they showed, and that information then systematically started to guide us as a community, as a scientific community, towards developing some therapeutic applications of that. Now here, for example, as you see, this is what I'm showing here is just a cell cycle. Body के अंदर cells are replicating. दिन के अंदर hundreds of thousands of new cells are built inside every body. As you're sitting here, those processes are going on, but this is the process which goes on inside the human body. So how the cell cycle works, how there is initiation, there is transformation of information, and then full cell is formed and then created, and that this cycle goes on and on. As our understanding of that became more and more clear, more and more drugs and better understanding of which part of the cell can be targeted in order to stop the recycle. For example, in case of cancer. So basically cancer cells, they progress or they replicate faster than the normal cells, and that's why it bada hota hai jaldi se, right? But if you know that about the cell cycle, then people will start thinking, let's try to at, let's say, the S state, G1 state, G2 state, can we understand more precisely and try to develop drugs which will hit only one state versus the other state. In any case, the idea is by doing so, you stop the cell cycle. If you stop the cell cycle, you stop the disease. So with that knowledge, more and more drugs started to come out. And I showed you earlier an example of paclitaxel, for example, right? You see up there, it's a mitotic spindle jo hai cell ka, it attacks there. That's one. So more and more, and so, you know, more drugs, as depending on what is the area of interest of the specific knowledge that we have and the area of interest of the people, those who are working on, they try to use that knowledge to target that part of the cell cycle. And that's how you will see various different drugs, even type of drugs, even classes of drugs named based on, you know, just for example, chemotherapy, or in this case, for example, DNA targeting drugs, right? You will hear the term immune therapy, which is more modern. Checkpoint inhibitor, what are checkpoints? So when you see when a one state, from S to G2 to M, in between these are checkpoints. So if can you target a checkpoint, so the process, so the cycle is stopped. So those became the checkpoint inhibitors. You hear those terms more commonly now in past couple of decades or so, because that's how immunologists are trying to just target that. So as our understanding develop, many drugs targeting like mTOR based, you know, apoptosis, autophagy, these are just processes within the cells which are being targeted by different drugs, and new therapeutic approaches have come out. So anyway, so the molecular mechanism underlying, you know, the anti-proliferative actions of, for example, paclitaxel, as I mentioned, it's a mitotic arrest. You have, I have given just a few of the names here. BCL2, you know, is that subsequently leads to apoptosis. Then again, there are drugs which are targeting, you know, different stages, checkpoint inhibitors. There are immune cells which promote an immune therapy, for example, because nature, for example, has built in the immune system. If the immune system is suppressed for whatever reason during the disease, due to disease, you develop a therapy which is only trying to break macrophages, for example. They suppress checkpoint CDK8, which are the immune system which helps to fight the disease. If you target those macrophages, and release the defense mechanism so it can fight based on the immune cells, that's immune therapy. So these various techniques and approaches have evolved over the years. So what I showed, for example, you know, initially in the mid 20th century, initially it was only observation based. People observed, based on that they did whatever they could at the time, and that led to, you know, the whatever known therapy at the time. As we learn more about organs, for example, about cells, so we start to use those cells to test molecules, right? So now cell is not a single entity, not a single macromolecule. It's a cluster of many proteins, but cell is a phenotype. So this is called phenotypic approach, phenotypic screening of the whole model, whether it's an animal model or patient-derived animal model. Then we learn based on the organic chemistry that you can design molecules, you can extract molecules, you can convert those into crystals or soluble form or into vapors, as inhalers, for example, are used. When more and more these techniques evolved, that was all in the 20th century. All of that. When up until, as I showed you, 1970s, there was a little bit more advanced organic chemistry, analytical chemistry, NMR, for example, was there, and you probably may know, you know what is magnetic resonance imaging, MRI. It came out of NMR. So NMR came first, and that application was then used to convert into the application which is MRI. Right? So that became more and more, so that in 70s, for example, it became more rational design of drugs. So you have, the understanding of the biological functions, the structural properties, and then based on those structural properties of the molecules, structural molecules of the small molecules to the properties of the biological molecules, or understanding of the nature of the biological molecules, you try to block the function or modify the function of biological molecules. That approach became in past 50 years or so, because that knowledge at the time wasn't there before that, and that knowledge became available, more and more understanding. So you take a much more so-called the rational approach. Now, what are these two different approaches? So as I mentioned, phenotypic screening. Phenotypic screen, for example, is like, for example, you have to screen organic molecules. You design organic molecules, or maybe you extract various molecules from nature, and you want to test those molecules against specific disease. Let's say in case, for example, a type of a cancer, right? So you take those cancer cells, the growth of the cancer cells, you can monitor, you can follow by treating different drugs and see that growth goes up and down, or the cells are killed, and that's it. So, but you have the whole cell that you are testing, whether the cell is affected or not. So, phenotypic screening. So you have whole phenotype against which we are trying to test your new drugs, and once you have that, you do. So in this approach, what happens is you have a cell, but in order to see whether your molecule is responding or not, you have to have an assay. What is the meaning of response? They should be a way of measuring, right? They should be a way of measuring. So that is an assay. If there are assays, so what you need is, for a phenotypic screening, you take a type of cell, and then you have an assay which is a reliable assay, is not misleading assay, but you have a good assay. You screen your compounds. Whatever unknown compounds that you have, you want to screen those, and then you identify the compounds which actually give you the effect that you are interested to see, so-called hits, initial compounds. Now those hits, now you want to identify that inside the cell, where actually are they hitting, which protein or which process. I showed you, in the previous slide, that cell cycle or whatever that may be. How is that small molecule affecting the growth of the cell? So you go hunting, analyzing, looking for what the target of that molecule is. So that's the process. The converse process, the other process which is used, is that you already know which target protein you want to target, right? So that is what is called the target-based drug discovery. So for example, I was previously, as I showed you, there's BCL2, let's say, for example, is one of the protein, P53, CMYC. They are proteins for which there's a lot of information already developed, known biology, and you want to target those molecules. So you have a biological target already in place. Then you develop an assay specifically for that target. So when you treat a compound, you treat with a compound of unknown compound, and if it reacts, let's say the function of the BCL2 or function of that protein is altered or suppressed, whatever your goal is, if you achieve that with the new molecules, you know that, okay, this molecule works. So you screen thousands of compounds, or whatever number of compounds you have, you screen those compounds and see which ones of those molecules actually react. You take those molecules, that becomes your hit then, right? And then you again develop assay, screen, identify hit. So you can get to the, now you want to take forward that information in order to see okay, what else can be done, right? So the question of what else can be done is now you are focusing, narrowing it down to specifically trying to understand the disease biology so that you can alter, you can modify, you can stop whatever your goal be. In some cases, drug is to activate. In case of cancer, for example, it could be for suppressing. So whatever that goal is, then you try to optimize that. As a chemist, for example, you try to optimize. So let's say the initial hit, you have the activity against your phenotypic screen, let's say you did, is at a micromolar range. You have to give certain micromolar concentration of the drug to get that response. Now that is very high. You want to narrow down, because if you convert that high, you convert that into a pill. If you do the calculation, there is a formula that is used. If you convert that, it will be a large pill, a lot of quantity that is to be. It's really not that effective. So pharmaceutical industry, when they try to do, is what they do is initially with the pharma industry or academia or wherever these kind of biotech or wherever these happens, you initially try to find out hits which are initially showing, giving you the signal that you're interested in, and that could be at maybe at a higher concentration. But then you start optimizing that. You try to, because your goal is that initially if it is giving a hit on the target, it could be anywhere, but it is close enough that you get some signal. Now you want to optimize that molecules, change its physiochemical character, keep on testing so it hits exactly here. The target in the center is just an indication. You want a compound that is at a low concentration, giving you the response that you need without giving you any toxic effect. Because if it gives you, it is, you know, it is a micromolar concentration if an initial compound is active, and you bring it down to nanomolar concentration, very small, or picomolar concentration, let's say, if you can really make that compound. If it is such a potent compound, great, but it should not be at the same time toxic for something else. Then it really cannot be used. So that is the process, and that process is actually is what the whole drug development thing is. You have HTS, is high throughput screening, the initial one, for example. You do that, you identify compound, you purify compound, then you start looking at whether it's soluble or not. Going back to your question, soluble or not is the reason is because if you take the molecule goes inside, but it is sitting inside, it is not going to the target, it will be just cleared out. It will not have therapeutic effect. It should be soluble so it can dissolve in plasma, it can get through whatever stream it is needed, you know, whether it's an oral route or if you take IV or whichever route, it should reach to the target where it should have the effect, intended effect. So then, so see, each stage you have to have in such a way that, you know, the biological property is maintained or optimized, the chemical property is optimized so that you have the biological target that you're looking for. So you develop an assay. No, assay could be various different forms of assay. Let's say enzymatic assay, either it's this stable bioassay. So you get whole, again, solubility issue. There is again, as you can see, each one of these is a discipline of science which gets involved. Once you have that, you know, enzymatic assay, you can have a cellular assay. Now the question is, if a molecule is needed to reach to a point, but if it cannot penetrate the cellular wall, it will not go through that. So even may be effective in your ex vivo assays or models, but if it is not going through, then it will not solve the purpose. If there is something which is needed, for example, neurological diseases, or neurological disease, whatever that disease is, it has to pass the blood-brain barrier. If the molecule be very effective, but it doesn't pass the blood-brain barrier, it's not going to affect. So that is another, as you can see, the criteria that has to be taken into account when you're developing. And then you have to have the right models to test into. Animal models should be relevant to what you want to see. in human. And now there were, the ex vivo human models, for example, are there, which is in last 20 plus years or so. PDX, patient-derived xenograft model, for example. You take the patient sample, you graft inside the animal, and you develop a model, and then you test your drug in that. But that's a technique. This is a very highly skilled art, because after some times, if you take the human cells, which are grafted inside the animal cells, after first passage, second passage, third passage, you have to know how many passages are needed in order, because the tumor microenvironment is actually of the environment that you are interested to see, because after some time the host will take over. So you have to know. So it's as you can see, this whole science has evolved in past around 25 years only, and now they are optimizing, making more skill. So as you can see, just making a molecule is not enough. Making a molecule is fine, but then optimizing based on your goal at each step is needed. Now here comes the question you are asking. Patte khaye ya extract liya. Look at this. Stomach tumor pH is 2. So when you take something orally, it straight goes to your gut. वहाँ का pH highly acidic. It breaks down everything. आपने टफ फूड खाया है वो, आपने लीव्स खाए हैं वो, आपने कुछ भी खाया है, पका हुआ खाना खाया है, कच्चा खाना खाया है, अंदर जाता है, pH 2 के अंदर कितनी चीज़ें सर्वाइव करेंगी? It breaks it down. So you have to, so that's in the stomach. When it gets to intestine, pH is slightly start to rise. So depending on how your molecule has to travel, you have to, and as you can see, it passes away, then the stability phase 1. Their pH is 3 to 8. There are many thousands of enzymes. Will the molecule survive there? Will it get there, or it will metabolize, it will break it down to something which is which therapeutic value, right? So it has to go through that. All of this process passes to the liver, enters into the blood, then you say if it has to get to the kidney, to renal, so it has to go through this many barriers, this whole journey it has to go through, right? So you have to have a very good understanding of each step of this biology. When it goes through all of this, the goal there is, now you see you started with initial property of a molecule, you found initially a hit somewhere there. And when you are optimizing the physiochemical properties, you are putting through all these chakravyus, and your molecule has to survive. If it does not pass any of these to the satisfactory level, drop it, start again, another molecule. So when you make a hit, you make various variations of the hits, you make various analogs. Each analog is being tested for all of this. So your ultimate goal is, it has to survive. Your ultimate goal is, you increase the property. Its activity should increase, but activity ke saath mein toxicity should not be there. Your goal is to bring it to that green, northeast growth. You want to bring your properties. So you can make a compound which is active, but uski properties theek nahi hai, niche hai, it will not survive. It will not become a drug. So as you can see here, it's a highly multidisciplinary complex enterprise to take something and convert it into a drug. But fortunately, as I showed you, that was the whole idea about when we go to the next lectures, you will see, to have the appreciation of what it takes to develop a drug. Simple painkiller जो कहते हैं, जो कि इतनी complicated disease नहीं होनी चाहिए, से लेके complicated disease, neurodegeneration, cancer, cardiovascular, infection जैसे, if nothing else, just imagine the complexity of human body. Coronavirus, right? It's a small virus जो नेकेड आई से नज़र भी नहीं आता, पूरी कायनात को बंद कर दिया था, पूरे वर्ल्ड को बंद कर दिया था. Nobody could move, right? So human body, how sensitive it is and how does it react, and also an eye opener कि ज़्यादा चालाक मत बनो, इतनी चीज़ भी तुमको हिला सकती है. There are many lessons to learn, but the point being that's a philosophical side, but the reality part is, and also actually biological side, to understand how complex the human body is, to understand what is actually a small thing, how it can have such a profound effect on the functions of human body, right? So because of all these complexities which are involved, so experimental therapeutics is actually techniques and methods and all the approaches, latest technologies, whatever that is, our know-how from various different disciplines, to bringing those together to develop a single drug for a disease, for any disease for that matter, which has some positive effect. It takes an army of people, army of brightest people to bring a drug. So next time when you see a drug, hopefully you don't take a drug, but when you see a drug, you should have an appreciation of a scientist, what it takes to bring a drug to the market. So I showed you, how it started from just observation based to more, as we learned about the science, more systematically the rational design, and then we learned more about genetics, genes, what are genes, what are proteins, the whole genome thing which came out about 20 years ago or so, understanding of what the genomes are, how genetic system, or how the gene and their functions, how the proteins, various parts of the, within the human body, and various macromolecules within the body, how they work, and that how best we can develop assays, how best we can understand the disease biology, led to more and more refined improvement in development of drugs. Any question before I go further? Yes. I'm sorry, what was what? That was just an example. You're talking about this. G1, what is G1 arrest? Apoptosis. Oh, aberrant G1 like. So G1 arrest is the G1 arrest, right? So arrest in before that, the proteins for which that you can, similar to that, you can just interfere in between there. So that's, well, I'm not going into the biology side. There's a huge biology behind it. That's aberrant G1 like, which is not G1 but G1 like steps which gets involved in there. So that's more of a biology. I'll have to give a whole biology lecture on that. But that's slightly different, but it looks it's similar to G1. Some form of that. Yeah, so for example, so two things. Two approaches. You're talking about these approach. I was trying to tell you two approaches. One is to approach the whole cell initially to understand that whether your new molecule gives a response or not. So that's it, targeting the cell. The other approach is inside the cell. If you know a function of a protein from that cycle, then you specifically target that protein. You develop your assays, you develop your approaches just to target that protein, because presumably you know that that protein has effect, has a major function in causing the disease or progression of the disease. So you specifically target that disease. So two different approaches. Yeah, if you know about the target, then you can take that approach to specifically target. But you can take parallel approach. You can do also phenotypic screening, or you can also do the target multiple approaches. For example, these targets that I mentioned have been known for decades and decades, and still we don't have a drug for that, target that, because obviously our understanding of the function, because as I mentioned earlier, that we are assuming that protein has a major function in progression. When you try to target that protein in the signaling pathway, something else is also happening. So, it's a domino effect. A triggers to B, B triggers to C, C triggers to D. You do not know the ultimate outcome of that is positive or negative. If it is positive, how much of it is positive? You may initially feel that you are targeting, but it immediately adjusts itself. So your initial assays may show that, okay, there is an effect, but after some time you see that it got adjusted. It didn't really have the outcome that you are looking for. So that is the reason. So yes, you can have two approaches, and these proteins have been known for a long time, but still we do not have a drug because they are very smart proteins. They adjust to any attack on them, and they still continue. Yeah, there are approaches being tried. Yes, the ultimate goal is whatever the target is, to reach the target. So yes, people are approaching this whole so-called nanotechnology or nano approach. This is basically one of an approach to get your drug to that specific target. Because if it doesn't reach to the target, it is not going to have an effect. And these, whether it is an vector approach, whether it is a nano approach, whether it is a simple, give one way of reaching to the target. These are various approaches to reach the enemy and attack the enemy. In this case, that specific protein that you presumably think is the cause of the disease. And if your molecule, if your missile is not getting there, these are the techniques that you're using to get there. So these are, again, as you can see, I'm calling loosely as techniques, but each one of these is a science. Each one is a discipline. Now, drug delivery, drug formulation itself is a huge field. But all of these fields are coming together and contributing to develop one approach which is effective for any disease of interest. Yeah. So let me repeat if I understood you correctly. So you said immune cells, right? So immune cells are being affected by immune therapy. हाँ. What is cancer? करेक्ट, ये तो बड़े ब्रॉड लेवल, मैक्रो लेवल पे है. माइक्रो लेवल पे व्हाट इज कैंसर? किसी को नहीं मालूम. इट्स ए क्लस्टर ऑफ डिजीजेस. इट्स ए क्लस्टर ऑफ वेरियस अप्रोचेस व्हिच आर कमिंग टुगेदर टू डू दैट. उसमें इम्यून सेल्स इज आल्सो पार्ट ऑफ दैट. जो आपने पूछा कि अगर इम्यून सेल्स भी इस तरह से हो रहा है, और इम्यून जैसे कार्टी जब आए, क्यूमेरिक एंटीजन रिसेप्टर टी सेल्स, जो अप्रोच आई जब कार्टी थेरेपी जो है, देयर आर 5 ड्रग्स व्हिच आर अप्रूव्ड नाउ, कार्टी थेरेपी जो है, जो उसका वन ऑफ द चैलेंज इज प्रिसाइजली व्हाट यू जस्ट आस्क्ड, कि दे grows. So CAR T approach, I'll come to that. So I'll not tell you now because I have a lecture which in the CAR T part is included. But there is that the control, they go out of control. एक जो theory about the COVID का था कि जो है what is called the kinetic burst जो होता है इतना growth उसका fast हो जाता है. So that is again a disease. You call it by different names, whatever name. So आपका question था कि cancer के अंदर भी. So yes, immune cells may also be the, you know, but then again if they are causing the progression of the growth का जो है progression is enhanced, it's cancer. So whether it's one type of cell or the other type of cell. Okay, so again to give you an appreciation of, this is again I'm telling you, you know, this sort of lecture, history and background, to give an appreciation of this, our knowledge, which is validated correction, which leads to a point which we can accept, okay, this is something acceptable. The speed with which our knowledge grows is relatively slow compared to our knowledge about the biology is slow compared to technology. Our understanding slowly is growing, and as we learn more and more, we try to develop new approaches. And with that, new and new, you know, the drugs, the approaches, the therapies, as we were just talking earlier, the immune therapy versus chemotherapy versus, you know, the different forms that were used historically were used. So this, for example, see, this microscope was developed in 1590. Still microscope is used, right? Techniques. But our biological understanding is slowly evolving. And here is an example of that. So previously, there was, you know, if there is, let's say, cancer, people will say lung cancer or breast cancer or ovarian cancer or lung cancer, you know, head and neck cancer. They were based on the organ because that was the understanding of. Now, within an organ, there could be different reasons, as I was telling earlier. The disease, the cause of disease for person one, for the same disease, the cause would be different, could be different for a person, next person, or many other people. So this understanding of really there are within the same disease type, there could be various different reasons, is very recent, and here's an example. So this is, I'm just showing you there was, you know, maybe now 10-15 years ago, there was some review done of 10,000 cancer patients, and this in this case only non-small cell lung cancer. And those patients, their causes of disease was analyzed, and there were at least 16 different proteins which were identified for non-small cell lung cancer. So as you can see, if a protein, one of these proteins, let's say KRAS or the EGFR, if in patient population which has MET as the cause of lung cancer, if a drug is developed to target MET, and if the patient cause of disease is MET, only then that drug will work, right? For this lung cancer patient, if the cause of disease is any one of different protein which is not MET, if you give the MET drug, it's not going to work because their target is different. So that is what led to now identifying, when a patient comes in, to identify, be able to analyze what is the protein cause for that patient. And then if there is a drug available, you try to give that drug for that specific target. And that is what is known as the personalized medicine. Now, because it is very person to person depending on, and if you assume that hopefully people or the doctor has enough tools in his or her toolbox to be able to offer to the patient to do that. So that is what led to, as you know, so those agents means drugs, will work only for that small subset of patient for which the target is the target of that agent. It will not work otherwise. So that is what has, now how do we know all of this? So as you can see, now you hear the term omics, proteomics, genomics, metabolomics, or all of these omics, right? As you see here, now go, just I'm trying to give you quickly the reason I gave this the history and background of the tool is just so they have an appreciation of what it takes to develop a drug. So as I mentioned, from simple solvent extraction to analyzing, in 1944 the drug, the quinine, was synthesized, while the cinchona based drug was observed, in bark effect was observed in 1747. It took 200 years to be able to synthesize the molecule in there, right? Then the analytical techniques were developed. Analytical chemistry, you know, this chromatography that we use, for example, and then led to HPLC is, usse pehle tha GC, gas chromatography, right? NMR came. Now there are, you know, even within chromatography, various forms of TLC, GC, LC, LCMS, mass spec. All these techniques have evolved, and all that has made it easier to identify and to understand what exactly the molecule is, whether it's a small molecule or biological molecule. That now these techniques have become more and more refined. So other than the small molecule, just the drug, you could also now learn about the biological molecules, and that is what led to, you know, understanding the genome, proteins, metabolites, transcriptomes, RNAs, and all that. And that's how, but the technique is the same. It's chromatography, but it is more and more refined with the mass spec and all of these things. And therefore you hear these terms, omics ki jo various terms that are used. Now see, with that our diagnostic power has increased. Now you can actually diagnose. If you take the patient sample, you can diagnose actually which protein is there. So you see, I gave you a history of going back to a few centuries, but our understanding of this is just Bisal. It's very recent. So if you look at the human scale, the speed with which science has evolved or evolving, and why it takes so long, because you see कि इतना टाइम हो गया, इतना खर्चा होता है, इतना billions of dollars जाते हैं, trillions of dollars जाते हैं, दवाइयाँ कहाँ हैं, right? Which is a fair question to ask. As a scientist, you should be thinking कि कितना complex है human body को समझना पहले, and there are not enough techniques available. हम चाहे चांद पे हो आए, मंगल पे हो आए, that is easier than understanding human body, human biology. So as more and more techniques are evolving, our understanding is improved. As our understanding is improved, we can diagnose hopefully better. We can diagnose which patient will respond to what drug, and that's how you can match. That's the personalized medicine, so to speak. But as you see, these you can ask these questions and you can hopefully answer these questions. Who is likely to develop a cancer? How would you know that? Because now you have ways to hopefully patterns are developed. I will come to in few slides, algorithm and AI based, machine learning based, all that. But those patterns are developed. Based on that, you can have prognostic analysis done. What type of cancer? Cancer भी है तो किस type का cancer है? उस cancer के अंदर भी, as I give you an example of non-small cell lung cancer, उस cancer के अंदर भी कौन सा actually protein है जो cause of the not other 15 but the one which is, उसके लिए you can create predictive marker कि you advance में पता चल जाए कि whether it's going to, you know, वो जो कहते हैं hereditary है, अगर वो genes निकला है, the rare diseases, if it is in the family, you can analyze it quickly, you can tell them कि भाई be cautious, अपनी जो है जो lifestyle change करो, अपना सब कुछ change करो, खाना पीना ठीक करो. So this is again based on, you can have predictive, but that all is collective information that came based on all the technologies that have developed in the past few decades. You get what is the optimal body weight, who will respond, what will not, will it come or will not it come. So you can ask these questions, and that is what is basically, next NGS, next generation sequencing. What it does is basically take the sample you analyze to ask all these questions, and based on that you can then stratify it, precision, okay, who will respond, how they will respond, are they the good candidate for what type of drug, whether the drug should be given or not, or some other combination should be given up. So all of these things became more and more effective in past couple of decades or so. But as you can see on the human time scale, the reason I wanted to keep these two lectures together is just to have an appreciation of, okay, so tomorrow if you are thinking of this, and frankly if you are talking to non-scientists, if these questions are asked, you can say, wait a minute, it's not as simple as it looks. It takes really, because we don't understand the human body and we don't have the techniques. Lighting chamakti hui to badi nazar aayenge, buildings badi nazar aayenge, you walk into 5-star malls and you will see all of this, but that has nothing to do. That's a very simple thing. Bahut choti cheez hai wo compared to human body, to understand the human body. So now as all of this is happening, so predictive knowledge, so you have probably be hearing for quite some time now what is known as the big data. Have you heard the terms big data and methods? So what basically is big data? So over the years, now think of, for example, I'll take a very simplified way. Let's say, for example, यहाँ पे AIMS है. Patients come in, उनके जो भी analysis किया जाता है, it gets recorded. I'm assuming it gets recorded. They go through treatment. Their pre-post treatment records are there. Multiply that with millions and millions of people just within AIMS, जो कि पिछले 100 साल में जब से बना है AIMS after independence, उतने सालों में, multiply that to how many AIMS or how many medical schools. I'm just using AIMS as just an example. How many different medical hospitals, schools, places. Same thing, think of animal studies, animal models created at IITs or Panjab universities or NIPERs or ISRs or jahan pe bhi, within only, I'm just giving reference to local, India ke saath mein. Now multiply that to each country having their own system. The world mein kitna data available hai. Human data se, preclinical data se leke sara available hai. उसको अगर स्टडी करने बैठे हैं, if you start looking at the data, if you start to analyze that data, there is so much information available, you can actually create patterns. For drug X in genetic pool that we study, let's say in case of India, for example, you take the genetic pool, whatever genetic pool is, people with that body mass, people with that age group, people with that lifestyle, people with that kind of their dietary habits, vegetarian, non-vegetarian, how much carb that they, if we keep track of all of those who responded or didn't respond, those who have the markers that could convert into a disease or not, if you have millions of that, a pattern will start developing. You'll say, okay, people in that age group, people whether their sex type, whether male or female or child or girl or a boy, age group, their sex type, their lifestyle, their dietary habits, they responded or didn't respond, एक pattern बनना शुरू होने लगता है. I'm giving you a very simplified version of that. Now multiply that world over if that data is available. If you start using and converting that data into useful information, what kind of markers were there, then now combine that to all the omics, let's say for example I mentioned. If there are samples available, you take those samples in those patient population, you do the testing, and you'll say, okay, these kind of proteins are actually emerging more and more for X type of population. Now you have used that data to convert into a very useful information, right? So that is what big data is. And data, and now see, how many different disciplines are needed. This data is being generated for decades and decades. Computers were not there. Data was being stored in whatever electronic form, before electronic, the paper form was there. But that knowledge was there. Now take all of that knowledge, computer storage capacity wasn't there, now you can talk about, take all of that data, big data, store it, try to create algorithm to understand and make meaningful outcome of those. It could save many years of research to see what kind of disease, what kind of drug should be developed, where people will respond or not, or who will be the responder or non-responder, right? That's all what is big data. I'm just using, for example, just giving an example of this gastroenterology and hepatology, for example. You take all of this information that is available, use omics, electronic health record, EHR, take that all patient reported data, clinical trials, you take all of that data and start to make sense out of that data. Okay, who responded, what worked, what didn't work, who will be the good candidate to study, who should not be studied because the drug is not going to make any help to them. It creates a useful information. So that's basically what the big data is. That is what the big data science is. And if you have all of that data available, that data could be very effective in trying to develop new therapies. So see, traditional ways, you look at the experimental, you do the discovery and go through the process that I just showed you in a few slides prior to this. If big data is available, then you can use that information, go through these experimental design, but now you know very targeted who will respond, and you can design your therapeutic approaches for a specific patient pool. So it narrows down the time, it saves money and effort, and hopefully more effective drugs can come out of that. So this is basically, you can take any one of these diseases, identify the biomarker based on, for example, if drugs are working in population, there are 10 million people, a drug is working in 500,000 people. You analyze based on omics उन 500,000 people के अंदर कौन सा प्रोटीन था जिसके ऊपर ये काम कर रहा है. Though you have a biomarker now, so for that biomarker now so it saved you a lot of time based on the prior data which was already available, and that can help to expedite the process. So this is again, the process that can be. So this is what basically artificial intelligence, AI. This is one of the applications that is being used, because now what basically you're doing is you have all these huge peak of the huge data available. You are writing basically algorithm to make sense of and create useful information to develop new therapies. So this is in summary basically what I just showed you is the journey of how it started, from where it was, and as you can see here, this is just last 15 years. It's just 15 years that what we are talking about, all these things. Okay, now, but still our understanding of this is again on the macro, outside of humans. Inside of, do we really understand how human cells work? It's a soup of, it's an ocean of knowledge, ocean of biology. So कहाँ गए उनका क्वेश्चन था, बनाने कुदरत की रचना बड़ी ज़बरदस्त है, उसको समझने के लिए आप लोगों ने बड़ा कंट्रीब्यूट करना है अब. So basically, this is the drug development process as you see here. Discover something, do preclinical, clinical development. वाला पार्ट तो अभी मैंने टच नहीं किया. I will come in one of those lectures. So this is the scientist which can be easily controlled. That's the regulatory, क्योंकि there should be control. अगर control नहीं हुआ, the people will just start using left and right, and that may have even further negative effects. So these are, we'll touch upon, we'll come back to that. This, how from preclinical, from discovery to clinical development that happens. So it's basically a team sport. And everybody has to pick up their load, everybody has to pick up their responsibility and contribute in order to get just one drug out or develop a therapy which is effective for the treatment of whatever the disease is, and to take care of the human health. With that, any questions? Yes? Any drug? It's coming. It's coming in my few lectures. So I'll touch upon that. There is, see, big data approach is very new, very recent. It may not necessarily give a drug, but it can contribute in developing something. If it can weed out, let's say 95% of the effort which will be wasted and say don't do this, it's a huge contribution. So there are a lot of approaches all around the world which were based on artificial intelligence, machine learning, AI, ML, you will see everywhere, and based on that trying to develop new therapies and approach. There is no currently a drug we can say this came out of because it's very recent. But it is definitely contributing tremendously in narrowing down the approach, narrowing down to the pool, which hopefully be more effective, faster, and with less resources. So, more to follow. Any other question? These approaches, there are many combination approaches being used. Again, how effective they are would require much better, much deeper understanding of the human biology. So initially, if you give something, it will give the reaction. It could be effective, the reaction that you're looking for. But then immediately the proteins adjust and goes back to the time. So it's a very short-term outcome that you will see. So, oh yes, combination approach is being tested for a long time and will continue because there are multiple approaches, because cell is not a simple thing, it's a very complex thing. Proteins, one has effect on the other. If you look at the structure of signaling pathways, for example, it's very hard to understand how one protein has an effect on another because they are not single, akele nahi khade, they are all connected. So therefore combination approaches were there, are being used, and there will probably be more and more so being used. But are there, how effective requires for us to better understand the disease biology and develop the right methods, the right models which can replicate what happens in human body. So ultimately these are being developed for human body. And you have to have model which is outside of human body which can tell you what will work in human body. You don't want to create humans as your guinea pig to test that. So that's where the regulatory things come into play. You can't just use left and right and start saying. So there are those complex, and we'll cover those as we go further. Thank you very much. Enjoy your afternoon.
Thank you so much, sir, for this wonderful lecture. We will now take a short lunch break, and all of you, please, we are going to resume the lecture after lunch. Please be here by 1:50 p.m. The water facility is right outside the door. If anyone needs to go for a water break, they can go outside. And also one more thing, the attendance will also be taken right after the lunch, so make sure you are all present here. Welcome back, everyone. Next up, I would like to request Dr. Aman Bhalla to deliver his lecture on asymmetric synthesis in drug designing.
A very good afternoon to all present. So, how many of students are from this department who done their MSc? Okay, 1 and 2. Okay. So, okay. So, the topic which I choose, that is asymmetric synthesis in drug designing. Okay. So, your main topic which your guest speaker is talking about all about the drugs. Okay. So, that's why I just taken this topic, so I can give you a brief about this topic. Okay. What is the need and what is the origin and how the requirement of this asymmetric synthesis. Okay. So, any student can explain me asymmetric synthesis? Any one of you? Any idea about it? Okay. Chirality generate karna. Okay. That answer is from our department. So that's good. Okay. So mainly in my all talks which you are going to take in the next session, I am not going to give you the deep about the mechanisms. Okay. I just want to know all of you that whenever any synthetic work has to be done, okay, you have to be very careful. You have to say that the molecule which you are getting, it should be pure. Okay, it should not be in a racemic form. So that's why we have to study asymmetric synthesis in drug designing. Okay, generally if we go to the starting, that is chirality. Everybody know from the +1 +2 level that we are talking about the chiral center, a carbon which is surrounded by 4 different groups. Okay. When you are talking about this molecule in 2 dimension, you can draw it easily, a carbon with 4 substituents. But when we are seeing this molecule in 3 dimension, when you are seeing this molecule or you are visualizing this molecule in 3 dimension, then you have to worry about it. Why you have to worry about it? Because if a molecule is surrounded by 4 different groups, you want to see the arrangement of that 4 different group in a space. Because in 2 dimension it is okay, you have to draw it. But in a 3 dimension, how you are looking to a molecule. For that reason, you have to see about the chirality and the enantiomeric access. Okay. So if we are just saying the chirality is what? It is a fundamental symmetry property of 3-dimensional objects. So generally हमने chirality की definition में क्या ध्यान में रखा हुआ? We are just talking about the mirror image. Okay. If you pick the molecule from one side and you try to place it on the other side, you are seeing whether it is imposed exactly or it is non-superimposed. So generally we say a chirality if it cannot be superimposed on its mirror image. Okay, this is a general trend which we are talking in our textbooks. We see a different type of pictures where we see what is chiral and what is achiral. So initial basic study tells us that if one part is non-superimposed on its other part, we called it as a chiral molecule. Okay. So if we are talking about the chirality, I am saying enantiomers is the term which come close to it. So can you explain what is enantiomer? If you are seeing a picture, in this molecule, your chiral carbon is surrounded by 4 different group. वो क्या है 4 different group? Methyl, hydrogen, COH and NH2. But if you are seeing the second picture, it is also the same thing. So what is the difference? If we have to see this in 2 dimension, we cannot say they are different. But if we have to see in 3 dimension, we have to find out the difference. But if you see the first line, a pair of isomer having identical physical and chemical property. Physical and chemical property क्यों change होगी? जब दोनों के पास groups और atoms of group same है. So that means they are similar. A pair of isomer having identical physical and chemical property. Now if you see second point, they rotate the plane polarized light. दोनों chiral molecules हैं. If you try to place in optical activity, they both rotate the plane polarized light. But now the difference start. But in opposite direction. Why? Why they are now in opposite direction? When a chiral carbon is surrounded by same 4 group, they have a same physical property, same chemical property, but why they are rotating the optical plane polarized light in opposite direction? From here onwards comes the picture of their arrangement in a space. So and their molecules are non-superimposable mirror image. So generally हम पूछते हैं what is enantiomer? Students लिख देते हैं they are non-superimposable mirror image of each other. We give half marks. First you have to describe the molecule. Molecule के atoms और groups of atoms same है. That means they have same chemical and physical property. अगर chemical and physical property same हो गई तो how you are going to get 2 different molecules? Then we move further that if we put them in an optical rotation experiment, they are rotating the plane polarized light in opposite direction. That means something is differentiating them. What is that differentiation? That means their arrangement. So if we are talking about the drugs, drug designing, how this can come into that? What is the need to study about this? Now just see a one picture which is in front of you. Okay, that is a common enzyme. Okay, I think everybody has done about the lock and key theory. Enzyme का पढ़ा है ना? Lock and key theory, fit induce theory, that you place a substrate, substrate will fit into the active sites of the enzyme. That is a simple concept. Now if you see the molecule, abhi in the morning lecture sir has given you one example of epinephrine. Okay, so if you just see the structure of this molecule, क्या difference है? In both structures the atom and group of atoms are similar. कोई change नहीं आया. Now try to look the chiral center. If you are looking the chiral center, वहाँ पे आपको एक change नजर आ रहा है. Can you see that change? The H and OH. Okay, वहाँ पे एक word लिखा हुआ है R and S. That is the configuration. Okay. Now if you see in the enzyme there are sites, anionic sites, flat area, and there is a blue site. So in first case, the OH molecule, which has the complementary interaction with respect to blue area that lie on the other side. So that molecule is not going to fit very actively. But if you see on the other side, the OH group of the molecule is going to fit tightly in the enzyme surface. So now how this differentiation is occurred? Only the arrangement of groups around the chiral carbon. So the world around us is chiral. Biological macromolecule of living system are chiral. Okay, that's why a drug molecule which is chiral has two enantiomers, thus will have a different interaction with the receptor site which is chiral also. Now what I want to conclude here, that if you have a same molecule, you didn't find the exact configuration, your molecule will not be approved by any drug agency. Now if you are able to find the correct configuration, your molecule will be further submitted for the next trials. Okay, so that's why different interaction lead to different biological effects of two enantiomers of a chiral drug. So if you are making any chiral drug molecule, you must know the exact configuration of that molecules. So that is the need. Okay, now this is the one example. If you see, have you listened about this molecule in history about thalidomide? इसके रिलेशन में आपने कभी कुछ सुना? इससे आवाज आ रही है? I can move. Have you listened about anything about this? Okay, a very generalized way. Now it's okay. In very generalized way, if we are not talking about any lecture, if we are not talking about any lecture, any interaction, just a reality, we have witnessed in last years about the COVID. We are the lucky one who have survived. Okay, but at that time, what was our major challenge? Will my immune system support? क्या मेरा सर्वाइवल होगा? That is the real challenge. I guess everybody has faced it. At that time, we don't have a accurate medicine which is required for that disease. So now guess, in a family, don't take it as any lecture program, any interaction, just a human being. That in a family, if we are expecting any new member in the family, what type of happiness we are surrounded. Okay, but when any drug is given to you or just as a pain reliever, but that drug lead to the deformities in the newly born child, how will you take it? For whole life it will remain as a special child. So in 1960 there is a one drug that is thalidomide. It gives to the women for the sedative. Okay, but that time it is a racemic mixture. Okay, so after the birth of the babies, many babies are with the different deformities. Now for the whole life they have to live as such. But if as a human being we can just sense that just taking one medicine can lead to the whole family in such a state, how much is the need to understand that if two enantiomers are there, one is causes fetal deformities, other is the only sedative. But that time they made thalidomide, it is a racemic mixture. Now what is the difference? As I told you in one example, what is the difference? Only arrangement. They have a similar physical properties, similar chemical properties. Okay. The only arrangement is different. So why to separate them? Just take a mixture, make a drug, use it. But can you imagine, can you sense that the one enantiomer is sedative only, the other enantiomer can lead to fetal deformities. So that is the major chunk, that asymmetric synthesis or knowledge of purity of one enantiomer is required, because second can lead to the major deformities. Okay. So it is not only regarding the 1960. Now can you see these two peoples? The Nobel Prize, I guess you all are from the science field, you understand what is Nobel Prize. It is something the extraordinary research, so then you can get the Nobel Prize. So it is only the asymmetric organocatalysis. It is in 2021. So my question is, my main topic is that it is not that asymmetric synthesis is in 1960, now there is no need. Still people are working in it and they are getting the Nobel Prize. Okay. So that is the need. Now the question is need because it has a very drastic effect. Now we know that if thalidomide is as a racemic mixture, no one in the world can going to use it. But if one of the enantiomer is in pure form, it can be used. Okay. So then the concept is, what these people done? They have development of new type of catalysis, asymmetric organocatalysis, that they give a catalyst which can provide only one enantiomer, not the other. Now try to figure out asymmetric synthesis. Okay. If you are planning to do a synthesis and by luck or by chance or by intention you are getting two type of enantiomers or diastereomers, what is your plan of action? Out of that enantiomer and diastereomer, one is required and other is not required. Then what is your take? What you want to do? Okay, if you have to synthesize the thalidomide, what is your plan? What do you want to synthesize? Out of these two, you want to synthesize one only in 100%. You do not want the second one. So here comes the requirement of asymmetric synthesis. That means it is not a symmetry wise, it is not the 50-50, it is asymmetry, that is on the basis of your requirement. Okay. So if you have to plan a asymmetric synthesis, then you must know that if there is a chiral carbon in the molecule, what is the arrangement? Okay. In thalidomide, if you go to the example of thalidomide, what is the difference? अगर आप थैलिमिडो के मॉलिक्यूल पे जाएंगे, कैन यू शो मी द डिफरेंस? टू मॉलिक्यूल्स में only about the chiral center. If you see the example of this molecule, it is also L-DOPA drug, which is used for anti-Parkinson. What is anti-Parkinson disease? Parkinson disease के बारे में सुना है? With age क्या होता है, क्या इफेक्ट आता है उसमें? नर्वस सिस्टम थोड़ा डाउन हो जाता है. क्लियर? सो इन दैट केस नाउ इन दोनों मॉलिक्यूल्स में आप अगर ध्यान से देखो, ओनली अरेंजमेंट का सिर्फ सिनेरियो है. अब कितना अच्छी बात है कि आपको ना तो कोई एटम और ग्रुप ऑफ एटम चेंज करना है, ना कोई नया मैकेनिज्म स्टेप इन्वॉल्व करना है, बट आपको क्या करना है, आपको रिएक्शन को ऐसे कंट्रोल करना है, यू विल गेट द वन enantiomer in majority in preference to the other one. So that is asymmetric synthesis in drug designing. So if any drug molecule which is chiral in nature, and many drugs which are available, they are chiral in nature, so they must be in a very pure form. So how this pure form is achieved, that we have to study. For first of all, you have to know the configuration. This is a tricky term which every time students get confused. Configuration and conformation. Can you differentiate between configuration and conformation? Anything come to your mind? Okay. When we are talking about the configuration, you have to pinpoint only at one carbon and what is the arrangement around it. Okay. एक कार्बन के आसपास कितने ग्रुप हो गए? 4. उसकी वैलेंसी 4 है तो 4 ग्रुप होंगे. So when you are seeing one carbon, there are 4 group around it. Okay. So 3 ग्रुप्स को आप प्लेन पे देख रहे हो, एक ग्रुप या तो बिलो द प्लेन होगा या अबव द प्लेन होगा, टुवर्ड्स यू होगा या आपसे दूर होगा. सो दैट इज कॉन्फ़िगरेशन, द सेपेटल अरेंजमेंट ऑफ एटम्स और ग्रुप्स ऑफ एटम्स अराउंड अ chiral कार्बन. तो कॉन्फ़िगरेशन कभी भी कंफ्यूज नहीं करना कॉन्फर्मेशन से. दैट कॉन्फ़िगरेशन, chiral carbon, and how the atom or group of atom are placed in space around this chiral carbon. Okay, so उसके set rules हैं. आपने R and S configuration, अब अगर आपकी आई, पहले आप देखते हो कि वो चारों ग्रुप कैसे प्रेजेंट है, आप उनकी प्रायोरिटी डिसाइड करते हो. प्रायोरिटी डिसाइड करने के बाद देखते हो कि आपकी आई कैसे ट्रैवल करती है. प्रायोरिटी वाइज जब उसको ट्रैवल करते हो क्लॉकवाइज, तो आप क्या देते हो, आर कॉन्फ़िगरेशन. अगर वो लेफ्ट साइड एंटी क्लॉकवाइज तो एस कॉन्फ़िगरेशन. सो दैट इज योर कॉन्फ़िगरेशन. नाउ कम्स टू द कॉन्फर्मेशन. नाउ इफ यू सी देयर इज अ मॉलिक्यूल ऑफ ईथेन, one carbon is jointed by another carbon. Now इन दोनों कार्बन पे अगर मैं 3 ग्रुप डिफरेंट लगा दूं तो दोनों कार्बन chiral हो जाएंगे. If I say in this group जो 2 हाइड्रोजन है, एक हाइड्रोजन में मैंने फ्लोरीन लगा दिया, सो उस कार्बन के आसपास क्या हुआ, फ्लोरीन, हाइड्रोजन एंड मिथाइल. So every carbon which is surrounded by 4 different group consist of configuration. Now मैंने एक कार्बन को फिक्स कर दिया, दूसरे कार्बन को रोटेट करना शुरू कर दिया. जब मैं दूसरे कार्बन को रोटेट कर रहा हूं, इधर ग्रुप्स की और इधर ग्रुप्स की आपस में जो अरेंजमेंट होती है, दैट इज कॉन्फर्मेशन. सो द क्वेश्चन है, इज configuration available in confirmation? Confirmation में configuration available होगी? Yes, because configuration is a different aspect. Any carbon which is surrounded by 4 different groups give you R and S, that is okay. But if you are talking about conformation, you have to fix one, then move the second, and then check the correlation. Okay, so in this way you have done, if this is the example of butane, you have done the anti-conformation, fully eclipsed, staggered, gauche, different type of configuration you have already studied in your. So this is the molecule of ethane. If you see staggered and eclipsed you have done, this is the conformation of butane, and if you see the different type of energies, and this is cyclohexane, इसमें आपने किया ना chair, boat, twist boat, half chair. Okay, so this is the concept which we are talking about. Now whenever we are going to asymmetric synthesis, chiral molecules, these two definition always stands. Okay, what is configuration, what is conformation? तो अब सबको इसका डिफरेंस क्लियर है. That is configuration with respect to particular carbon, and conformation is when you fix one, change the second, what is the relation between both of them, that is your conformation. Okay, so for that we have to move to the chiral. Okay, if there is a one chiral center, you are going to get two different type of molecules, whether it is R configuration and S configuration. If you have a more chiral center, then what is the possible stereoisomer? That is the, it depend upon the number of chiral center. Okay, so till now you know what is chirality, what is configuration, what is conformation. अब if we are talking about a stereogenic unit, तो जब आप ड्रग मॉलिक्यूल की सिंथेसिस का प्लान कर रहे हैं, यू हैव टू नो हाउ माई ड्रग मॉलिक्यूल इज अ chiral, और वट एवर द मॉलिक्यूल यू आर प्रिपेयरिंग, हाउ इट इज chiral, what is the unit in it which define it that this molecule is chiral in nature. So you have to check the different stereogenic units present in it. This we already done, but if you just look at this picture, simple example, your X is surrounded by A, B, C, D. There are 4 different groups surrounded, so that is your centrally chiral molecule, because chirality start from the center. Center is your X. Okay, now if you see in this molecule, whenever you are talking about a double bond का एग्जाम्पल क्या हो सकता है? कार्बन कार्बन डबल बॉन्ड, कार्बोनाइल डबल बॉन्ड, they are always in the plane, they have a planar conformation sp2. So if you are talking about a plane, then the groups which are present above the plane or below the plane. So if you are moving, this is your carbon carbon double bond, carbon oxygen double bond. Okay, now in this case, if you are talking about the chirality, okay, you move from this axis. Okay, now the groups are lying above, groups are lying below. In that case, you can say my molecule has a axially chiral, because chirality is defined from the axis. Okay, so now if you see that your molecule has a plane, some groups are above and some groups are below, then you can say it is the planar chirality. So first of all, you have to define your molecule has a central chirality, axial chirality or planar chirality. Okay, so now if you know about this chirality, you have to check that your molecule has one chiral center and two chiral center. So if it is a one chiral center, you call it enantiomer. Enantiomer की definition की थी. Okay, now if your molecule has two generally stand for dia. Okay, now see in this picture, what is the difference? वो हम करेंगे ऑन द बेसिस ऑफ एक्सटर्नल सोर्स इफ वी आर अप्लाइंग. They are going to differentiate when any external source is going to apply. Okay, that I will explain that how they are going to differentiate. Okay, so if you see the first chiral carbon and in the second picture, can you see that is the mirror image? First chiral carbon. Now move to the second chiral carbon, they have the same configuration. Okay, so in diastereomers that they are not exactly the known superimposable mirror image of each other. The first example that you have to study, dia means 2. 2 means there must be 2 chiral center present in it. Okay, so whenever any example you are saying, you have to see that whether it has a 1 chiral center and the 2 chiral center. Okay, in drug molecule, if you are talking about the anti-cancer Taxol type molecules, they have more than 8-9 chiral centers. Okay, we are just talking about only 1 and 2 chiral centers. So that's why you have to know about this enantiomers, diastereomers. Now can you see in this both have 2 chiral center, but if you cut this molecule from the plane, the first molecule exactly lie to the second molecule. So that is your plane of symmetry. Okay, so if you mix these both enantiomers, we call it as a racemic mixture. Okay, so this all terminologies which I just give a brief about is they all come into this slide. So this is the only slide that if you are planning any drug molecules in the synthesis, everybody must know this. And this is important. Why? Because why asymmetric synthesis? Number 1, often only one enantiomer is biological active. अब आपने enantiomer एक chiral center से आपके 2 enantiomer बनेंगे, 2 chiral center से आपके 4 different molecules बनेंगे. आपकी requirement तो सिर्फ एक है जो अच्छे से activity देगा, rest 3 are going to interfere. So often only one enantiomer is biological active. Now can you just feel it? अगर आपका सेकेंड enantiomer भी पार्ट हुआ, क्या ड्रास्टिकल चेंज लेके आ सकता है? कैन यू रियलाइज द थैलिमिडो केस वेयर प्रेजेंस ऑफ ओनली वन enantiomer can lead to the deformities? So that's why only one enantiomer is biological active. What is the second question? Both enantiomer may have a different activity. I have prepared a drug molecule, I get 2 enantiomers. One enantiomer is antibacterial in nature, other enantiomer is antiviral in nature, anti-inflammatory, anti-cancer, anything, because in 3 dimension they are interacting with the enzymes in a different way. Okay, so both enantiomers may have different activity. Third query क्या हो सकती है? One may be desired, the other is inactive and undesired. आपको एक enantiomer चाहिए, दूसरा नहीं चाहिए, वो undesired हो सकता है. जो undesired enantiomer है, that can lead to the environmental pollution. So if you see, sometimes optical active enantiomer is twice as active as compared to racemic mixture. अगर आप यहाँ पे स्क्रीन में एक एग्जाम्पल देखोगे, इस एग्जाम्पल में आपके पास सिंपल एलिफेटिक चेन है और एक 5 membered रिंग है. Now there are 2 important aspects in this. डबल बॉन्ड है। अगर आपके पास डबल बॉन्ड हो तो आप ज्योमेट्रिकल आइसोमेरिज्म पॉसिबल कर सकते हो, E और Z कर सकते हो अकॉर्डिंग टू अरेंजमेंट ऑफ ग्रुप्स अराउंड द कार्बन कार्बन डबल बॉन्ड। सेकेंड, आप इसमें एक कायरल सेंटर भी देख रहे हो। सो इफ यू सी इन फर्स्ट केस your chiral center has a configuration R and your double bond has a configuration Z. Okay? अब ये आपने एक मॉलिक्यूल सिंपल लैब में आपको सिंथेसाइज करने के लिए दिया, दिस इज अ इंसेक्ट फेरेमोन। ओके, ये आपको सिंथेसाइज करने के लिए दिया, आपने बहुत एफिशिएंटली सिंथेसाइज किया, 99% आपने एसिमेट्रिक सिंथेसिस अचीव कर ली इसमें। कैन यू सी presence of 1% of S and Z isomer can inhibit activity completely. So if a synthesis has been planned for such type of molecule, you are still getting the 99% one enantiomer in a pure form, the other 1% completely inhibit the activity of the 99%. So such is the requirement. Now the question comes, why? How we people survived in COVID? Because our immune system responded differently. Some have not survived. Unfortunately, their immune system responded differently. But the virus is same. COVID virus is same for all. So this is the difference, that we don't know that how our immune system is working with respect to outer antigens. So if we are getting a drug which is 99% pure, my immune system is a very good one. So that 1% is not going to check, but someone is allergic to that thing. So presence of that 1% can lead to the drastic effect to that human being. So that's why if we are talking about the enantiomeric purity, if you check, everybody, I pray that no one should take medicine, but if you have taken, if you check the backside of the, there is every percentage wise purity has been written, that is FDA or drug, गवर्नमेंट ऑफ इंडिया की जो ड्रग की इंस्टीट्यूशन है, दे अप्रूव इट ऑन द बेसिस ऑफ दैट परसेंटेज. Okay, so the requirement that comes is only with respect to this. Now I am just, you don't have to remember anything, you don't have to just see what is the mechanism, just see the change and the difference. Now in this case nothing is going to change in anything of the structure, atom, group, bonding, nothing, only the arrangement in 2 dimensions. So I am saying, if you are drawing this molecule in your register, you are not going to see any change, but if you seeing in 3 dimension, just see the properties which they are showing. In first molecule, if you see, that is beta blocker for heart diseases, second is contraceptive. In below you see, one is no narcotic activity, the other is showing the narcotic activity. Now these molecules are different only in the arrangement. If you see the example of aspartame, you have a 4 different type of enantiomers and diastereomers which are possible, but out of this 4 only one is sweet. And the most latest example, that is of penicillin. The one molecule is antibiotic, the other molecule is inactive. Now can you explain me what is the difference between first and second? क्या difference आपको first and second में लग रहा है? Their phenyl, CH2, CO, NH bond, they are same, 4 member cycling ring. The only difference is, if you see carbon 3 and carbon 4, the presence of hydrogen atoms. In first case, if you see the arrangement of these two hydrogens with respect to each other, it is cis. Now in the next molecule, if you see the hydrogen, they are trans. Now can you see such a big molecule, only hydrogen atom which is just arranging whether it is on the same side and whether it is on the opposite side. It completely make the molecule active and inactive. So penicillin is the first antibiotic drug which nature has helped us to reach for that molecule. Now if you see the difference between penicillin and epipenicillin, it is just arrangement of hydrogens. Now if you see other chirality is also present, that is a bicyclic molecule, carbonyl group, amide group, all are present, but the difference is only single hydrogen atoms, whether they are cis or trans. That difference is only can lead to the formation of one is antibiotic and other is not antibiotic. So if you are planning to synthesize any such type of molecule and you are not seeing the configuration in a molecule, then you are able to synthesize a new molecule. You can do the molecular docking study, you can do the ADMI study, but when you try to put these molecules for biological activity, whether you are getting or not, it is a different aspect. Why? Because the appropriate configuration is required to hook with the molecules which are present in the living system. ये जो ड्रग मॉलिक्यूल है, जो सुबह उन्होंने आपसे ड्रग की डेफिनेशन पूछी थी, ये जो ड्रग मॉलिक्यूल्स है वो कहाँ यूज करने वाले हैं? वी आर नॉट पुट इन द ग्लास बॉटल एंड कीप इन द होम. उसको कोई सजा के रखने के लिए तो नहीं लाता ना. दैट इज फॉर द लिविंग बीइंग. सो वो लिविंग बीइंग से कैसे रिएक्ट करेंगे, अगर उसकी थोड़ा सा भी बेसिस माइंड में नहीं रखा एंड जस्ट ट्राई टू प्रिपेयर अ मॉलिक्यूल, देन इट विल बी वेरी डिफरेंट एस्पेक्ट्स. ओके, सो दैट इज लास्ट वन, दैट इज we are starting asymmetric synthesis. Even when the other enantiomer is inert, it is still desirable to synthesize and use the active enantiomer only in pure form. So what I am talking about is asymmetric synthesis, that if you are getting more than one enantiomer, firstly you have to separate them, you have to purify them. Now it is a common practice that any new molecule, if it is in the market, you have to give the all the enantiomers, diastereomers, all the purity consent, then it can be put into the market. Okay, so we will continue tomorrow from here. Okay, sir, बढ़िया.
Thank you so much, sir, for this amazing lecture. Next up, I would like to request Professor Gurjaspreet Singh to share a few words about our guest, Professor Narinder Singh.
Good afternoon to all of you. So, Professor Narinder Singh is a distinguished faculty member in the Department of Chemistry at the Indian Institute of Technology, IIT Ropar. He earned his PhD from Guru Nanak Dev University, Amritsar, and went on to pursue postdoctoral research in Japan, South Korea, UK, and before joining IIT Ropar in 2009. Professor Singh is known for his pioneer work in the field of supramolecular chemistry, material science, and chemical sensing. His research focuses on the design and development of fluorescent turn on chemosensor, particularly using calyx for arenes, tripodal frameworks, ionic liquids, and nanoparticle based system for the detection of environmentally and biologically relevant ions. He has significant contribution to lifestyle imaging, environment monitoring, and drug delivery research. With over 350 publications and more than 9,000 citations. He holds an impressive H-index of 45. His work has been featured in prestigious journals like JACS, JOC, Organic Letter, Talenta. Behind its research, he is a dedicated mentor to PhD scholars and actively involved in several national and interdisciplinary projects. His contributions reflect not only academic excellency but also a strong commitment to applied science for societal and strategic benefit. So now I invite Professor Ninder to deliver his talk.
Yeah, thank you very much for the invitation. And my work, 50% work is concerned with the chemosensors and some other work for the applied application. Today, I am not going to talk about chemosensor. So my today's talk is about antimicrobial agent and their biomedical application and environmental application. Especially, we will talk about the surface engineering, means how to make the membranes, how to make the antimicrobial surfaces, how to make a self-cleaning surfaces. So those things we will discuss today. So those who are new to my presentation, so for them that I am mostly involved for the chemosensor and especially for it's not biosensor, only chemosensor for determine the toxin in soil and water. And once we know that okay, this industrial water is contaminated with one particular type of toxin, then we make the method for removal of those toxins. And that is also a water treatment. And some of the material which are the waste in the industry be converted into useful entities, and several of our technologies are being used in the industry. Today I will talk about the water treatment as well as self-cleaning surfaces. So if I talk about antimicrobial activities or self-cleaning surfaces, so AMR, first thing is AMR, that is antimicrobial resistance. What happens? Now, many people they are taking the drug molecule and some of the animal as well as poultry, every day they give small amount of antibiotic to those animals. And after that, those type of toxins, they go to the water bodies. And if there are n number of bacteria, and if you put first antimicrobial agent, it will kill some of the bacteria. But those bacteria which are resistant to that drug, then they will propagate like anything. Now, if the next drug will come, then out of those population, some will be killed and other will be resistant to the two drug. In this way, there is a generation of multi-drug resistant bacteria. So multi-drug resistant bacteria or AMR, antimicrobial resistance, it's a great concern of today's scientific community. And if we see that 4.95 million people in 2019, they were affected with this type of MDR bacteria. Even you may have realized that sometimes somebody is having skin infection, and after that you are treating with one antibiotic, another antibiotic, again you are not get rid of that type of skin infection, probably because of the resistant fungus is there in the body. Especially those type of things are happen in the toes of the forefoot. So those type of things are there. So some of the anti or bacterial disease include like pneumonia and other type of disease are there. So now if I see the history of antimicrobial drug, so first period was the pre-antibiotic era when the penicillin was invented. You know, for penicillin, Fleming, he get the Nobel Prize, and after that there was a golden age when if there is any problem, they give the antibiotic agent, immediately they kill the material or kill the bacteria. But in the year of 1948, there was some of the bacteria which become resistant for the penicillin. Even there is another bacteria invented in India that is, in the penicillin we have lactam ring, right? We have a lactam ring. And in India, in New Delhi, and name is also New Delhi metallo-beta lactamase enzyme. That enzyme is secreted by the bacteria. It will break the lactam ring. If it will break the lactam ring, it means penicillin or anything which is derived from the penicillin G, that will not come into the effect to the as antibiotic are concerned. So that's why again it will be a resistant variety. So after that there was a era when the things happen me too drug means a large number of drugs were synthesized. Not only this beta lactam based drug, there are large number of other drugs which were synthesized, and after that they realized that some of around 60 antibiotics were released into the market. Cephalosporin is one, carbapenem is one. So those type of drugs are the example which was invented in this year. And after that there is a discovery void, means if I see the literature between 2001 to today, there are hardly any drug which was coming to the market and which was invented and after that it is coming to the market. Most of the time repurposing of drug is happening, and we also never take that type of risk to make a new drug for the market. Actually we have also combined some of the drugs using supramolecular interaction. Let us say if I have one drug, if I have two drugs, if I combine in such a way that after that they will not lose their chemical identity, but they become such a material that they will give some synergetic effect, then it will be a reasonably good progress. So those type of things are going on at the moment, and I will discuss those things today also. So if I have to see the drug resistant bacteria MRSA, actually I have also worked for MRSA. In fact, we have invented some of the compounds which are active against the MRSA. That is S. aureus, it becomes a resistant for S. aureus, it's a resistant variety. If you see, India is also, although more than 80% means red color, orange color means around 60 to 70% area of India is also affected with this MRSA. And there is also another drug that is carbapenem. There are bacteria which are resistant to the carbapenem, and that is quite predominant in the Indian states. And there is another third generation resistant varieties are also there in the India. Even fluoroquinolones based drugs, they are also resistant in the India. So means there are large number of bacterial colonies which are there in the country, for which we have to address the problem by giving a new direction for the bacteria. If I see the literature, earlier literature, if you see, then at that time this beta lactam ring was there, be it is carbapenem, be it is cephalosporin, and after that there is a generation of some of the antimicrobial peptides are there in the literature. Some people made self-penetrating peptide so that they will enter to the microbes, and after that they will cause some of the antimicrobial activities. And after that, with the peptides, there is one problem. One is the synthesis of peptide, that is a major problem. Another problem with peptide is you have to keep at a ambient temperature, and if there are unregulated amount of any salt, then peptide most of the time they will coagulate or they will lose their activity. So that's why after that people those who are working in the area of organic chemistry, they thought let us combine some of the features of those type of peptides in the form of peptidomimetic. So basically peptidomimetic means looking almost similar to the peptide. So they made the peptidomimetic like that way. And person who are belonging to my community means supramolecular scientist, we try to synthesize those type of molecules. So we were thinking what are the application or what are the affinities which we are getting from this antimicrobial agents. So basically it's a supramolecular assembly, means there is a some are hydrogen bond donor, some are the hydrogen bond acceptor. So when this type of molecule are there in the literature, peptidomimetic based compound were there in the literature which are offering too much hydrogen bond donor site, too much hydrogen bond acceptor site. Then the people like me, we thought let us try our fate with the molecule who are good hydrogen bond donor, who are good hydrogen bond acceptor, and top on that let us put some of the molecule like heterocyclic moiety. Let us see what is happening with this type of molecule. So that is another generation of material which are there in the literature. And top on that some of the metal line like silver, you know that it's a good antibacterial agent, and copper. During corona period we have also invented some of the formulation for antibacterial activity using copper nanocluster, which were eventually patented, and that technology was transferred to the industry, and it is today it is commercially available in the market, which was invented in my lab. That was the copper nanocluster based things. So I mean to say that there are large number of strategy by which you can attain the antimicrobial properties out of the system. So how those antibiotics, how they are working? Either first thing is either they will break the cell wall of bacteria, and if the cell wall will break down, then protoplasm will come outside from that bacteria. Another thing is there is a flux pump, means whatsoever waste material is there, bacteria get rid of that one. So some of the drug affect that pump. So that's why again toxin accumulate in the protoplasm of bacteria. And another is they are interacting with the cellular enzyme. Enzyme has to do some work, and if there are some of the drug molecule which interact with those type of enzyme, and after that it is affecting the activity of enzyme, automatically it will disturb the normal functioning of bacteria, and some are inhibiting by in the protein biosynthesis. So large number of mechanisms are there. So my method of working is like that way. At least you attack at least on two sides. Either I work with the membrane along with the inhibition of enzyme, or I will work with something it will combine with, or it will interact with the DNA, as well as it is affecting this flux pump. So I am try to combine two approaches together in my drug molecules. So we will discuss that later on. So now if we see peptides, linear peptide are there in the literature, which have shown excellent antibacterial activity, and some of the molecules which we have having that type of tryptophan like of moiety, that are also extensive in the literature along with the guanidine salt. And another is again peptide molecule. So if I see peptide or peptidomimetic type of thing, I have noticed one thing, that either that tryptophan like fluorophore or tryptophan is there, another is guanidine is there. So I thought let us combine these structure feature in the new group of our compounds. So and first is what is the benefit of this type of peptide? If you ask me that okay, there are large number of peptide, I have shown over here why peptide are so fascinating and what activity I will get out of these peptides. So activity I will get in that way. Either those peptide, let us say this is the cell membrane of that, let us say this is the bacteria and this is the cell wall of that bacteria. Either these peptide will create this type of barrel shape model, means it will create a hole and from where protoplasm will come out, or it will create the storipod pore, or carpet model, means this is the bacterial cell wall, something is attaching here, something attaching here, something here, something here. After that whenever there will be bacterial fission from that area where we have that carpet model, the protoplasm will come out, and after that it will create the bacterial cell death. So although there are some of the limitation, we know peptide has limitation. First limitation is it's a peptide, it's a amide bond is there, and if there is a pepsin type of enzyme, tryptophan is there, those type of enzymes are there, immediately protein will degrade and peptide will degrade, and and after that if my peptide will degrade, then it means it will not give the activity whatsoever I am anticipating from that molecules. Another is cytotoxicity. Some of the peptide, they are cytotoxic, like snake venom. It's a one type of protein, but it is toxic. Lack of oral bioavailability, these things, this I will take little later, and resistance to the bacteria. Bacteria become very resistant to this type of peptide. There are mechanism, I will not go to that detail, and production cost is too much high. If I have to generate a peptide molecule having 4 or 5 amino acid and synthetically I have to commercialize it, it is very expensive business. So that is a big hurdle. And another problem is the stability of that formulation. As I said, that if there is an interaction of, if there is a high salt strength, if there is a alteration in the pH, immediately my peptide molecule will break down. So means stability of formulation is a big issue. To counter all these things, now before going to the design of my molecule, let me show you that most of the time if we have zinc oxide, we have silver, we have copper oxide, they are acting as a antimicrobial agent, as I have shown over there. After that, what is happening? They lyse the bacteria, and after that protoplasm is coming out. So now you see how we design our receptor. So we have used the combined approach. First is amino acid. We have seen extensively we have seen in the literature that amino acid are there in the peptides, in the molecule who are successful to give the antimicrobial activity, and urea and thiourea derivatives are also there. Another thing is we have seen either people have combined as metal or metal oxide nanoparticle, and another thing is long chain should be there, long alkyl chain. If you can provide long alkyl chain, it's also quite good. So those type of things are there. So we combine to all these things together in a molecule, and after that, top on that, we self-assemble that molecule. And those self-assembled molecule we have investigated in the form of organogel, in the form of mycel, in the form of hydrogel, to see that which supramolecular assembly is giving better effect. So this was our design. You can see over there. More better we can show you. If we go by the organic synthesis, so we have started with tryptophan, and after that we combine with one of the pyridine carboxylic acid we have used, 2 pyridine carboxylic acid along with the methyl derivative of tryptophan. Methyl derivative we have taken in for such a way that after that we want to do the amide coupling by using some of the amide coupling reagent like HBTU, and that reagent are not working if you have the ester. So ester protection we have given over there, and this ester protection is better in the next step. After that we can treat it with the hydrogen hydrate, and we will create another amine over there. So pyridine we have taken. If I take the alkyl bromide, long chain alkyl bromide, if I do the alkylation over there, it will give me another set of receptors which we have commercialized recently. And after that, if you do the next reaction, we will get the urea derivative. So now you see the design. Tryptophan we have, peptide like structure we have, urea thiourea based receptor we have. So then one of the example, in this way you can synthesize the urea derivative. So we have taken one example. I can show you the results of that example. So first what we did, we made the hydrogel with this one. Why hydrogel? Those who are from the medical background, they know, maybe they are aware that if I have only one photosystem, it's not working. And if I have a group of photosynthesis, photosystems, then it will work like anything in the photosynthesis. So same approach we have utilized over there. Let us see what happen if we have only that single organic molecule. What will happen if we have self-assembled receptor? What will happen if we decorate with silver nanoparticle or copper nanoparticle? And what will happen if we take the zero gel out of this organogel? So let us combine, means I mean to say that it's not like that way you synthesize n number of compound, 15 compound bana lijiye, or after that you claim okay only one is working. No, it's fine. But on the other hand, you have another approach, means you make one compound and try to generate large number of supramolecular assemblies from that one compound. Let us see which type of merit we are getting out of it. So this is the organogel. When if we have this compound, and after that if you try to make the organogel, so you can see we will get the nice pattern of self-assembled structure. And after that, if you dry the sample, you will get the zero gel. Again in the zero gel, those supramolecular assemblies, they are not disturbed. It's not like that that way, okay, whenever we are having some water, only then it will give you a supramolecular assembly. No, it's not like that way. If you dry that water, again zero gel is generated, which is also having very good supramolecular interactions. And after that, if I interact with the silver or copper, then the shape of those organogels, this is being altered, and this is changed from a bundle shape to the either this is cluster type or this is a fiber type. So those type of shapes has been changed. And if I generate the zero gel out of these two, whatsoever organogel silver I have, organogel copper I have, if I generate the zero gel out of that, almost the similar type of shape or similar type of geometry I am getting. So means supramolecular interaction. Again, if I take only zero gel, if I take only organogel, if I take organogel with copper, organogel with silver, their supramolecular interaction are not disturbed if I remove the water out of the system. So is there any molecular level interaction? Yes, we have molecular level interaction. RP4 means we have a pure molecule. You can see carbonyl group is 1703, and whenever it is converted to the organogel, carbonyl group is shifted. It means there are some interaction in which this carbonyl group is participating. And again, when we interact with the silver, those carbonyl group are responsible to interact with silver or copper. So means some reasonable supramolecular interactions are there. And if you see the comparison of our compound, that is pure compound, organogel and organogel silver nanoparticle, clearly we can show that whenever we have compound, it was not acting as a very good bacterial viability is quite high, means it's not giving you any antibacterial activity. But the molecule is converted to organogel, you can see the zone and you can see the reduction. Reduction is remarkable. So I mean to say, if you have any compound, it is not giving you the activity, convert it into some entity, some self-assembled structure, maybe you will get the better structure, or maybe you will get the better activity. And after that, if I put little bit of silver nanoparticle, again the activity is being approved. It's not only for E. coli we have checked with another bacteria, that is S. Same pattern we have seen over there. And this is the zone of inhibition, as I have said earlier. The previous was for the silver nanoparticle, even for the copper nanoparticle, same trend we have seen with the copper nanoparticle. And if I have to compare between copper and the silver, then this this data is there in the form of zone inhibition. You can see in any case, copper is not better than silver, but it is reasonably good than that of only organogel in both the cases. And if we see the MIC, minimal inhibitory concentration, again that is from organogel to copper, organogel copper, it is giving the much better result for as far as MIC is concerned. Now let us see what will happen if I make the zero gel. Means if I compare a blank organogel, blank means only receptor, organogel, organogel copper, organogel silver, in the form of zero-gel and organogel. You can see organogel is little bit better than the zero-gel. But with organogel, you have to make it stable. That is the one problem which you have to take into account. So in that way, we have seen over there that otherwise zero gel is also giving you some of the activity which is not expected otherwise from the silver or copper nanoparticle. Now another work we have started, that is chitosan based hydrogel. We have started with. Why chitosan? Because it's a biodegradable, biocompatible. People saying it's non-antigenic, non-toxic. So large number of these type of activity or large number of these type of properties are there in the chitosan. So we thought, instead of making the hydrogel with our molecule, what will happen if we take the some application from or some activity from our molecule mixed with chitosan? Let us see is there any improvement. So accordingly, in the literature we have seen that there are large number of reports where people utilize their molecule along with chitosan, and after that they have seen some of the improvement. So again we have taken another molecule RP05, and in this molecule we have utilized along with the silver nanoparticle glutaraldehyde, that is a cross link, and we have generated some of the hydrogels, and those hydrogels shape you can see the for hydrogel like that way, and after that some characterization, and after that we have seen that they are also having very good activity, antibacterial activity. Somehow data I don't have at the moment, recently we did in that way. So you can make the hydrogel if from the supramolecular assembly of pure molecule, if you are facing some problem, you can utilize chitosan type of molecule. That also gives you a very good activity. Another type of molecule which in fact we popularize in the country, that is organic cation. If you see over there, those are the organic benzimidazolium, imidazolium. I have 4 or 5 patents which I commercialize on the basis of imidazolium and benzimidazolium based antibacterial agents. So long back we have synthesized this type of compound. Now there are some iron sulfur bacteria, and those bacteria need iron for their metabolism, and if we give that type of molecule which get rid of iron from the bacteria, then it will kill the bacteria. So this work was concerned with that type of activities. Another thing, like long chain, you can attach your molecule in that way, where we have let us see. So if we have silver, you can coat your molecule on the surface of silver, and after that you can create a self-cleaning surfaces, antimicrobial surfaces you can create. Long back we have done this work. Same type of material on one side you can attach with some entity like CNT you can put, and after that you can give organic cation, and long chain you can give, and you will expect a different type of activity out of this one. So very recently, this is the work. Previous work was like that way. For example, this is the surface. You apply that material, it will create a self-cleaning surface. So recently, we try to work for air purification. What we did, we have taken electrospin fiber and mixed with our compound, and after that we put in a system. There was one type of gadget, like this way gadget is there. Inside polluted air was there. Here our filter was there. From this side we are exposing all the air to the media, and after that we were checking that how much growth is coming to that media, and recently we published that it's an excellent antimicrobial or electrospin nanofiber which are having very good antibacterial or antioxidant activity for air purification. So this is one part, that is how to achieve the antimicrobial activity. Another part is to get rid of AMR, means antimicrobial resistance. We have made very large number of membranes. If you pass the water from that membrane, after that the water will be purified as far as your bacteria is concerned. So recently we published one work on nitrate removal, and this membrane is further modified for the bacterial removal. So what was the synthesis? So as title of my presentation was organic cation. What are those organic cation? If you take benzimidazole, react with this, either you can take bromoacetic acid or ester of bromoacetic acid, so you will get that type of positive charge on the nitrogen. Now you see, if you see the whatsoever lysol you are using at your house, the active ingredient in lysol is BKC, benzalkonium chloride, or in most of the cases the other are DDAC. You know? So means positively charged nitrogen is there, or quaternary ammonium salt is there. So that is acting as a antibacterial activity. So we try to give that type of antibacterial activity from that nitrogen, and after that this molecule get reacted with the hydrogen hydrate, and next reaction amine formation we generated this compound. So now let us see the activity of this compound. So when this compound was interacting with the iron oxide, after that we got some of the nanoparticle, iron oxide based nanoparticle. We actually, our first work was that there is a water sample. We try to suspend this material in the water sample, and after that once we get the antimicrobial activity, we want to recover this material by using the magnet. This was the thought process for us to utilize this material where we are getting, we are putting iron oxide as a material with which we can remove this entity from the water bodies. However, that was not as per our desire, work was not progressing in that direction. So then we thought, let us put on the silica, and after that let us make a heterogeneous catalyst. Let us see that those activities. So basically the purpose was like that way. We suspend our material in the water body, and after its activity we recover our material with the magnet. That was the thought process, but somehow it gave a different type of property. So if we see the SEM of iron oxide, silica along with our molecule, so those type of SEMs shows that small particle are there, and after that when it was interacting with nitrate, there was a quenching in the quenching in the fluorescence intensity, which clearly show that nitrate is binding with this entity. Then we thought, let us change our direction from antimicrobial to nitrate removal. So for that we made the membrane, polyimide based membrane we made. You can see in those membranes that there are small perforation is there, so that if water is coming, it will pass through the membrane, but most of the things will interact with the membrane, so that nitrate will interact, or nitrate will restrict on the membrane. And if I compare I and J, you can see in that region where I and J is there, and after interaction there are large amount of nitrate which is present on the membrane after utilization. There is another application with this membrane. If you shine light, if it's a pure membrane, and after that if you utilize it for long, after that you can see there was a change in the emission intensity. So means your membrane is now full with the nitrate or not, you will get to know only from light shining, only by shining the light. So removal you can see, and after that saturation factor we have calculated. If the water is containing less amount of nitrate, obviously the saturation factor is quite high. Otherwise, if you have high amount of nitrate, very easily you will polarize the membrane, and after that that membrane will not be of much use. So means in this membrane we have tried to combine or try to remove the nitrate exclusively, and after activity the color of that membrane was also changing. Another work we have recently did for the removal of antibiotic, means as I said, there are two applications. Either I have to generate new drugs which will give a antimicrobial activity better than the existing in the literature. Another way of addressing the things, means our antibiotic should not go to the environment, so that they will not create any type of antimicrobial resistance. So that work was inspired from that idea, that let us make some of the membrane which remove the antibiotic from the wastewater. So in that work what we did, we have taken organic nanoparticle, our material, and after that we made the metal complex, cobalt complex we made, and after that we made the membrane out of this, and eventually we find that it's removing the amoxicillin from the wastewater or from the household water. Receptor is very simple. If you see just in 1 hour you can synthesize in your lab this receptor. Purposefully we have kept nitrogen, that amino group as free, because when we will synthesize the polyamide, basically it's a polysulfonate membrane over here, but another membrane which is being used in the commercial production from our group which was invented, there we utilize triacid diamine along with this receptor, so that covalently this compound will attach to the membrane. And if you see upon successive addition of cobalt, there was a change in the absorption. It means cobalt is binding by utilizing 2 nitrogen, one is sp3 nitrogen, sp2 nitrogen and the hydroxyl group. When the cobalt will bind over there, it will affect the ring current of azodide. So means after that there is a modulation or there is a change in the band gap. So that's why we are getting the shift. And if we see the particle size, you can see that that is coming less than 1 micron in size. So if it is less than 1 micron, very easily I can put it in the membrane having size more than 1 micron. So after that we try to interact with the amoxicillin. It was working like anything, it was working for the amoxicillin. So then I had found region to made antimicrobial or not antimicrobial membranes for the separation of antibiotics. So organic nanoparticle you can see like that way. After interacting with cobalt, it was agglomerating to some extent, but with the amoxicillin it was agglomerating more, and then you can see the nice structure of our membrane. You can see we have controlled the porosity to an appreciable extent. Actually we did lot of work in the membrane technology, and the day I got this SEM with one of my students, she was working for this one. So I was very happy then when she said that now we can control the thickness of membrane, we can also control the porosity. So nice pores are there, so that water can only percolate out of that one and other things will restrict over there. Now this is the lower membrane, C membrane, which was utilized before that amoxicillin, and after that when amoxicillin is combining, you can see that type of agglomerated structure are there, which we could see on the surface of the membrane. Again our membrane is giving 2 results. One is they are purifying the amoxicillin. Another thing, there is a change in the color. You can see initially our membrane was like orange type of color, and if we continue to utilize for amoxicillin, after that when the whole membrane is covered with amoxicillin, it will convert to the, you can see the color will be light yellow type of color. So means if membrane is provided to you, it's a light yellow, it means it is already saturated with the amoxicillin. So these 2 applications we have shown with our membrane. Another approach is, third approach is also there. You can ask me that okay, membrane is there, so whenever there is a high level of antibiotic amoxicillin, all the time it will polarize the membrane, all the time you have to buy new membrane. So but then we thought, let us make another strategy, let us combine 2 things together, like I mean to say another way, let us say there is a 1 cup of tea containing some tea. So and there are some mosquitoes there. So what we have to do, we have to first pass through the sieve, so that इसमें से पत्ती निकल जाए ना. After that you can whatsoever application you want from that tea you can do. So means if I combine the step 2 in that way, now before going that one, let me show you another thing why we are doing that strategy. Now you can see here, if in the water I have low concentration of nitrate, my membrane will last long for a longer time. If I am having high concentration, then my membrane will not last long to an appreciable time. Now my strategy is, let us put some of the chemical which first precipitate most of the amoxicillin, and after that whenever there is a small amount of that antibiotic, after that utilize the membrane, then our membrane will last long for longer time. So in that intention, we made some of the things. Let us do those type of things. Pre-concentration by self-assembly for aminoglycosides based antibiotics. So we have started with that antibiotics. So like one of my students, he said in that way. So I have put the same example over here. He said that let us say there are large number of animals. If one animal will try to go from this door, every animal can go outside. But if together they try to go outside, then it will be difficult for them to go out. So in the same way, what we did, if we have only antibiotic agent, and that antibiotic agent or antibiotic material interacting with our molecule, maybe this assembly become a water soluble. So if it will self-assemble in that way, definitely it will precipitate down. So that was the guideline principle in our mind at that time. So accordingly, I said, okay, let us make that type of compound where we put some of the chromophore, we put hydrophobic moiety, we put hydrogen bond donor sites, we put hydrogen bond acceptor site, and neomycin we have utilized. In the neomycin again we have hydrogen bond donor, hydrogen bond acceptor. If you want to know the synthesis, so we take N protected amino acid, and after that we react with the protected phenylalanine, and we got the product. TFA is utilized to break the box, otherwise it was a box protector. To break that box we use the TFA, and after that we did this amine linkages synthesis, we got the final product. Now directly you will see, if I show you over here, after that when we were interacting with that one, material was precipitating in that one, in that vial. Immediately it was precipitating, and after that if we redisperse in the buffer, you can redisperse in the buffer in that way. So what was happening, let us see in that way systematically. Otherwise the organic nanoparticle or self-assembled structure was having size less than 0.1 micron, and when the neomycin was interacting, it was showing small interaction, not very large interaction it was showing. However, maybe I missed one, yeah, this one. So those nanoparticle, and after that when it come in contact with the neomycin, after that we got those type of agglomerated structure in that way we have shown diagrammatically over there. So, and if you put buffer solution, you can resuspend in the solution. So means agglomeration is there. So first you do the agglomeration of your material. Now the level of antibiotics will be lesser in that water sample. Now you can pass through from the membrane, and after that your membrane will also saved, and you can also achieve the target whatsoever you are designing. Another thing is, recently we have started with supramolecular solvent. It's a new thing, maybe it will be a new thing for some of the students. So means we have some of the solvent which act as a supramolecular end. Otherwise most of the time solvent are not interacting with your molecule. So in fact recently one of our review article, it published last week, where we have shown that eutectic solvent are acting as the supramolecular solvent. What are those supramolecular solvent? Basically these solvent are interacting with the drug, and after that they separate the drug out of the system. So how they are working, these supramolecular solvents? So in the supramolecular solvent, we make the micelle, and in that micelle we put our compound. You can see that compound with one pot synthesis we generated the compound which you can see over there. We put that compound. Purposefully we have put that compound because it has pyrene ring, it has, and in the drug molecule we also have highly lipophilic character we have, and that lipophilic character is interacting with this pyrene, and after that whenever there is a pi-pi interaction, which we can evaluate that pi-pi interaction from fluorescence spectroscopy, after that everything was precipitating out. So in that way it was precipitating out, so it was giving me the fact whatsoever I was expecting from that one, means some amount of AMT is separating from the solution. So that is another approach which we have utilized. So third approach which recently we utilized, that is organogels for the detection and decontamination of thiabendazole, those type of drug molecule. Again here we are utilizing, basically we had a very small molecule. In the previous example, our molecule was quite large and it was expensive to synthesize. Here we have taken small molecule. By using terbium, we convert it into the supramolecular assembly. In that way, we made the gel, and you can see in that gel, we remain successful to show that it remain in the form of fiber. Metallogel is formed in the form of fiber, and those fiber, when they were interacting with the, now terbium is there, so that's why that type of fluorescence you can see out of that terbium, and upon interaction there was a change in the fluorescence intensity. So mechanism, you know why we are getting this type of pattern. Basically we are getting the phosphorescence with the terbium. Last approach, peptidomimetic based system for the removal of pharmaceutical ingredient from the ETP. Basically we had one project from Baddi, from the ETP, CETP, where we were working for the removal of API from the wastewater. Then and this student was working for us, and he excited out of this one. He said, let me publish this work. I said, okay, you publish. So again we had a very small amount of very small peptide which was interacting with the silver. Now you know silver gives the linear complexes. If you have two pyridine, it will give you the linear complexes, and if it will give linear complexes, after that you have hydrogen bond donor, hydrogen bond acceptor. So you will get a nice type of pattern like this type of, and the molecule when antibiotic are interacting, immediately it become a lipophilic entities. So CETP, in CETP what is happening, if this is a pharmaceutical industry, first waste is coming to the coagulation tank, where you add the coagulating agent, and some of the material coagulate, some undergo flocculation, and after that there is a settler tank which is also sedimentation unit, and in that sedimentation unit you will get the sludge which is recirculated to the aerobic chamber if you have, otherwise it will go to the aerobic as well as anaerobic chamber. In anaerobic there is a HRT, hydraulic retention time is of 12 days, in anaerobic it's a 3 day, but after all these activity, even if there is a small amount of API, it will reduce your BOD, COD, but if there is a pharmaceutical ingredient, it's very difficult to get rid of that one. In fact, that pharmaceutical ingredient kills the bacteria in anaerobic as well as an aerobic chamber. So that's why this whatsoever water, treated water is coming from the system, although it is meeting the standards, that is BOD less than 30, COD less than 240, TDS less than 2100, but small amount of pharmaceutical content is there. That's why most of the time regulatory impose high penalty. So then we made a strategy. After all this treatment, I said, you take this type of fibril structure, after all this treatment, interact your treated water through, we have fixed bed of chemical engineering ke hain ko bacche nahi. So basically there are various type of system by which you can do the filtration. It can be fixed bed, it can be floating bed, it can be fluidized bed. Here we utilize the fixed bed strategy, and you pass the water sample out of that one, and after that we have seen that there was no amount of antibiotic in the water sample. So compound structure was like that, supramolecular assembly with silver was like that, and after that when it was interacting you can see some of the changes in the photophysical property, which we have even shown with the proton NMR. And we can also make some gel type of material out of this one, and after that you can apply that gel on the membrane. So by 2-3 different type of strategy we can have to utilize this material. So with this I think I have addressed what are antimicrobial, how to address that resistance. So we have couple of minutes if you have any.
Slide number
38 ji. Yeah, 38 enter.
antioxidant air purification वो
अच्छा जी, अच्छा अच्छा
सब न्यू स्क्रीन लगी है पता नहीं
हाँ जी
न्यू स्क्रीन लगी है ना कर लिए
अच्छा 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 कोई नहीं लैग आ थोड़ा जिहा ये कौन दा ये हुन जरा ज़्यादा ही अगे लंघ गया वैसे तो 38 एंटर करना तो आना चाहिए हैगा 38 एंटर आ लो जी आ गया हुन आ गया हाँ जी for example we have this much size one material ਤੇ ਉਹਦੇ ਵਿੱਚ उसके अंदर हमने ये रख दिया यहाँ पर बैक्टीरिया for example we have some dish containing lot of bacteria and now there was one pipe and here we had membrane, this membrane. So after that we have given air flux from this side and we have collected air from here and directly that air was poured to the water and that was water was utilized for on the media plate. So if there will be any bacteria then it will grow on the media plate. So from there we have calculated okay it is having antibacterial activity but antioxidant activity is there in the fiber. If you take the fiber, you put in the antioxidant activity in the assay of antioxidant, so you will see that okay this fiber has some antioxidant activity.
And next to next slide go proton की property formation of carbon हाँ ये this one so H H Br नहीं है base में Br negative and positive H कहाँ
नहीं
reactant का
वो बड़ा
हाँ जी
नहीं वो basic medium है
अच्छा
hydrogen hydrate is a basic medium and after that you will get
first step
पहले
base
वाला
नहीं डाला वहाँ कोई भी एस्ट्रोनाइट्राइल 24 आवर इंटीरियर फ्लेक्स ओनली
हाँ हाँ नहीं नहीं ਉਹਦੇ ਵਿੱਚੋਂ HBr ਤੋਂ ਬਾਹਰ ਨਿਕਲਦਾ ਫॉर ਐਗਜ਼ਾਮਪਲ ਦੋ ਪਾਸੇ ਲੱਗਣਗੇ ਨਾ
अच्छा एक HBr बाहर चलेगा
ਇੱਕ ਜਿਹੜਾ ਇੱਕ ਪਾਸੇ ਤਾਂ Br ਆ ਗਿਆ ਦੂਜਾ HBr ਆ ਗਿਆ
और आपका बेस कोई नहीं पाया HBr
नहीं ਇਹਦੇ ਵਿੱਚ ਬੇਸ ਨਹੀਂ ਪਾਉਂਦੇ ਐਕਚੁਅਲੀ this sp2 nitrogen it has quite good LNP ਉਹ ਇਹਦਾ ਰਿਐਕਸ਼ਨ ਵੀ ਬਹੁਤ ਵਧੀਆ ਹੈਗੀ you take in the acetonitrile put these two together and reflux for 5-6 hours you will get white precipitate out of this
Any other question? Any question? Yes,
thank you
We have a tea break for 10 minutes and we'll resume the tutorial after that. Thank you. ले ली ले ली
Okay, better now? Sorry, yeah. Any question? Anything you want to ask from this morning? Are there people, there were more faces this morning. Are there people outside we were waiting for, or? उन्होंने कहा आज की दिहाड़ी हो गई. No, it's fine. So what I have done is, this is all for you guys. So what I will do is, what I have prepared this, I will be, based on each lecture, I have taken many of the, so before coming, पिछले कई हफ्तों से I was just trying to see which will be the best article after each lecture that you can, as a reference to additional information. So I made this folder for you, and what I'm going to do is I will give it to Dr. Gurjaspreet Singh or someone to make copies, or also maybe I'll share with him soft copies, so he can share whichever way he like. But so there are, इसमें अभी मैं जब lunch break के बाद में I was just going through these, इसमें से 2-3 हैं which are relevant to what we talked early in the morning. So anyway, I'll have, I think for all lectures, at least the ones that I'll be giving, I have related material that they are referenced in the latest publications or review articles that you can use as a reference, जो भी जो आपका interest है करते हैं. But that will give you a lot more कि latest क्या हो रहा है in each of these technologies, areas, discussion जो भी है. Okay. Thanks. So just to overview, okay, so we talk about, you'll see the way I have sequence of lectures is such, okay, because I was told mostly chemistry focused, chemistry based students, you do not get exposure to the biology side or you know, things which are especially experimental therapeutics, chemical biology and all of that. So you'll see the way I have designed the lecture, they are in such a way that you will see the connectivity going from one to another, so that when you see the whole picture in front of you, you'll say, कुछ तो काम का था. You see, so that's why I have, you'll see. So abhi jo next, intentionally I have given the title for this, the bridging chemical, you know, bringing chemical solutions to biological problems. So chemist, chemical solution, tube light upar ho jati hai, okay, this we know. So I'll try to connect to the biological side, you know, how and what is useful and how rest of the other fields than chemistry and how their applications and why their applications. So I'll be going up and down, make them basic fundamentals that you have seen, but ultimate goal is to connect you के biological side में उसका क्या application है. So if you look at a single unit, so remember I'll show you one of my slides again. I told you, to me, as I learned, periodic table is the building block of life, right? So think, for example, from atoms to molecules to cell to tissue to organ systems. This is how the complexity starts. So if we understand the fundamental, कि पहली ईंट जो है उसके गुण क्या हैं, there are ईंट, imagine all those periodic table के जो हैं, उनकी अपनी individual properties क्या है. If you understand that, then we can brick by brick, you can build the castle, right? So basically what that is, we know. So I'll quickly run through, which you probably may have already, you know, I'm pretty sure आपके earlier preliminary में लिया होगा, but as I said, my goal is to connect you to the biological side. Okay, take chemistry, take all of these things to connect to the fundamentals biological side. So we know that atoms क्या है? I mean, we know that each element has a nucleus, has orbits, electrons, protons, depending on what that is. And as we know in the periodic table, the groups, the grouping, group 1, group 2, all of that is based on the number of electrons and protons which are outside. So we know that they have those properties. Those properties, you know, proton, neutrons, each one has that, positive, negative, you know. So going back to fundamental, you know that 1s2, 2s2, 2p, right? Then comes 3s2, 3p2, 4, d orbital, right? So these orbitals make it. Each one has their property. So building from there, you know, they connect, whether they connect or they don't connect. Again, depending on what their properties are, which one is, you know, hydrogen has single electron, helium is a neutral gas, right? Helium, neon, उस periodic table, उस group में जितने भी हैं. So all of these, they have properties, and we can take advantage of their properties in order to build the blocks by blocks. So these, we know that helium with oxygen not going to work, hydrogen with oxygen marks the living molecule, water. So these properties have been now, and that is simply giving, how protons, neutrons, unki properties is leading to an element. And that is what leads to eventually to this, right? Periodic table, each जो है column, वो जो neutral gases, we started non-reactive, highly reactive, because they have one electron in the outer shell, right? And then their properties, depending on what their polarity, which is from here, if you go to fluorine, the halogens are the most, right? Because they need only one electron to complete their octet. So these properties actually constitute the bigger atoms. So now, as you'll probably next time when you see periodic table, you say, "Oh, life aa gayi," so to speak. Because these properties make the molecules. All these are combination. Now, there was another course that I was teaching a few years ago, and then I did some analysis. Physical existence. There are 5 atoms or 6 atoms in this periodic table. They are the most abundant rachnakar ki rachna mein. If you think of Earth, for example, hydrogen, nitrogen, carbon, oxygen, phosphorus, sulfur. Rest of those are in terms of, if you take the quantitative weight, if you take the Earth ka jo weight hai, including all the biological creation on the Earth, these 6 atoms are the ones which are more prevalent, and among these also, again I will repeat, ये creation की रचना है. Hydrogen, oxygen, 70% of the body weight is water, about H2O, right? Nitrogen, your amino acids. Without amino acids, you don't have oxygen, carbon. So NH2 on one side, or NH3 on one side, or COH on the other side. कितने हुए? NH, C and O. अब जितने मर्जी आप प्रोटीन उसमें अमीनो एसिड बना लें, आप ऊपर नीचे ऊपर को डेकोरेट ही करना है. उन अमीनो एसिड ने पेप्टाइड बनाने हैं. Those peptide will make the proteins will make the RNAs and the DNAs, right? Atoms तो चाहिए हैं इस periodic table में, those are contributing. Think of all the biological creation. Then of course we have other, I'm talking about by weight. So the entire Earth का जो weight है, you can literally can see there are few molecules that actually constitute the larger biological creation. चाहे आप पत्ते देख लें, चाहे animals देख लें, चाहे humans देख लें. जो creation है, biological creation is around 6 or 8 of those, 6 more so, these atoms. And this all permutation combination. आप मैंने कहा अब इसके बाद में आप सोच के देखना जब आप कभी भी आपके सामने जब आए. Anything that you look at, even if you look at the structure of DNA, or if you look at protein or simple amino acids, or simple water, simple alcohol, जो कि नहीं पीना चाहिए. So you look at all of these things, you will see that there are only a handful of atoms which constitute that. So if you can understand the properties of those, you can better understand जो आपका बड़ा goal है, which is a bigger macromolecule, and then applications of macromolecules. They are so when I say understanding, how they are made, what their reactivity is, and how that reactivity has positive or negative effect. So घूम फिर के फिर वही आ गया chemistry, but understanding of that part is important. So now that is again slowly, you look at this for example. So simple atom. If you look at water, Earth, if you look at 70%, give or take 70% is what? Oceans. पानी है. Again those same creation. उस पानी के कारण ही ऑक्सीजन है. ऑक्सीजन की कितना लेवल जाता है कि इतने एनिमल्स जो हैं, जो एक्वाटिक एनिमल्स हैं, जीते हैं. हम ऊपर जो अर्थ के ऊपर रहने वाले एनिमल्स हैं उससे जीते करते हैं. So understanding that part may will have help. You look at carbon, we know their properties. 4 valencies, 4 atoms. Take that to bigger atoms. So if we understand how these reactivity is happening, how much they can react, what they can react, you see where I'm going with this? It will help us to understand how the peptides, उनकी reactivity कैसे design, how you can design new peptides, or how the peptides react depending on what is their bells and whistles. क्या substituents हैं, what their structures is, what their properties are, or you can manipulate their physiochemical property by designing new one, just put a reactive group on one end, put another reactive group in the middle, put reactive group somewhere else, you can make a peptide which is reactive. You can keep on changing, you can play as a chemist, you have the advantage of, but the advantage comes by knowing the basic fundamental, knowing the properties of those things. But you see how knowing these properties now helps in biological molecules. So bringing chemical solutions to biological part is again understanding the chemical part side of it. So see, I have, as you can see, this will be absolutely fundamental जो मैं आपको दिखा रहा हूँ. It's not that something I'm telling you is new. You know all these things. Already you've seen this thing. But how, and then again, as we know the periodic table, as I just simply mentioned one point, you know, halogens जो हैं, fluorine, chlorine, bromine, iodine, जितने उस periodic table, उस column में हैं, their reactivity is different, right? Same way, different atoms, different columns. So now we can play with their reactivity. Most polar ionic potential, the highest ionic potential कौन सी atom की है? Fluorine. Same number of atoms but in a very compact, less orbitals, compact है 1s2 2s2 2p6, right? So उसमें 1s2 2s2 and then 2p5 एक ही चाहिए. तो कम स्पेस में 9 एटम्स 9 इलेक्ट्रॉन्स है. जैसे वो बढ़ता जाता है उसकी डेंसिटी पर साइज कम होती जाती है तो उसकी आयनिक पोटेंशियल चेंज होती जाती है. So knowing that thing, so the reason I specifically picked up fluorine is because fluorine you will see in the structure of many drugs. Also because fluorine is a small atom, compact small atom, the structures में इसलिए drugs में आता है कि maybe its property is such it can enter the biological pocket of the protein and can enter and go to the target site. So you can now design your drugs by introducing different atoms, playing and changing the physiochemical properties of the atoms. But again, going back to, if you have to solve a biological problem, we need to know what the solution, chemical solution can offer. So again, very fundamental. Now, if you have sodium chloride, right? Sodium chloride, simple molecule, right? Salt, sodium plus chlorine. एक के पास एक कम है, एक के पास एक ज़्यादा है, दोनों की फ्रेंडशिप है, they are highly reactive, right? Sodium has one extra electron, chlorine needs one electron, forms an electronic bond, ionic bonds, and that is how. And just imagine, salt नहीं होते तो क्या होता? हमारे taste का क्या होता? हमारे खानों का क्या होता? But that's one side of it. Sodium channel inside the human body, balancing. जो hypertension, BP higher, the first thing doctor will say, okay, नमक कम कर दो. So that छोटा सा atom क्यों इतना important है? Why is that so important? कहते जी नमक कम कर दो आप थोड़ा, क्योंकि biological आपका जो cardio या जो neurological कुछ है, because your sodium channel, potassium channel जो है, there is something based on what we know. So, but as a chemist, knowing the property, what it can do, right? It gives you advantage. So now based on just few examples that I gave, think of now how the reactions are inside the human body. थोड़ी गर्मी लगती है. We are in summer weather. If you stand outside, प्यास लगने लगती है. Dehydration होता है. In remote areas, stand longer, जो स्ट्रेट धूप लगती है तो कल उसको लू ना लग जाए बिकॉज डिहाइड्रेशन और सारा है ज़्यादा, राइट? उसको रिवर्स करने के लिए you hydrate yourself, right? There are enzymes inside which do this. So simple क्या है hydrolysis? Breakdown of molecules using water. And an enzyme की जो activity है, the hydrolases as group of enzymes are in many parts of the body. Different cells have that. Their functions vary from where they are depending on that, but hydrolases are very common, right? And so knowing those things, now you understand what the chemical reactions are, you understand the chemical properties are, and you understand how their reactions are important, their functions are important inside the human body. So let's expand even further. So अब जो चीज़ें पीनी खानी है, this is a very simplified version of few things which we regularly consume, right? अब देखिए उसका जो है, if you look at what is on the acidic side, which is on the basic side, उसका consumption जो है, if you take those things inside, I give you simply one example of simply hydrolysis. I haven't talked about other enzymes. Simple, because hydrolysis is a very common reaction inside the human body. But see what happens from or unka pH, whatever their pH is of the items that we take. Now look on the right side, pH in various parts of the body. So, but in terms of reaction, I just talked about only hydrolysis. But how much just you see that simple reaction or simple eating, we may call it simple, but how complex all the machinery is going on just based on the hydrolysis reaction alone. You see how much difference it makes. So unka jo hai, understanding as a chemist or understanding the properties of those chemical reactions of those atoms and chemical reactions inside this is the most complex chemical machinery ever made, is human body. बाकी की chemical industry तो जो इंसान ने जितना समझ आया बनाने की कोशिश की है, ये जो है ये chemical industry है, it's a very big chemical plant. From every step, आप brain से लेके अपने foot तक देखिए, हर चीज़ में कोई ना कोई chemical reaction हो रही है, whether it's a pure chemical reaction, but this is again influenced by enzymes. So these are enzymatic but chemical reactions which are happening. So understanding how various these things that we intake, things which affect various parts of the body, is basically chemical reactions. So if we understand those, if you understand simple reaction, let's look at this for example. Now, biological buffers, acetate is a common thing which is used, right? These buffers, basically what they do, a protein and acidic side पे एक hydrogen निकलता है that binds, it satisfies the buffer के साथ, given back and take enzymatic reaction होती है. And then again, if you look at that hydronic acid, again simple hydrogen in give and take, breaks it down to carbon dioxide and water. It's an enzymatic reaction that happened. But just imagine when you feel that you have acidity, right? Heartburn, quote unquote heartburn, bicarbonate soda लिया और पी लिया, था क्या? Is simply sodium bicarbonate. Simple sodium bicarbonate mixed in water. वही तो है, दो मॉलिक्यूल है. But that mix inside the water, the moment you take it, it breaks it down, it creates water and gas, it releases whatever is stuck up. आपको डकार आते हैं या जो भी होता है, that pressure is released. But what happened? It was a simple reaction that happened. So be able to understand how these things happen, how it works. We take advantage of trying to create solutions for those. So look at now biological molecules. So क्या है? I mean, if you look at the biological polymer molecules, if we look at, so let me take you to the next. There are 4 classes of molecules. You know all of those. I'm just bringing those together. Carbohydrates, sugar, and different forms of carbohydrates. So हम लेते हैं different type of glucose, sugar, sucrose, mannose, maltose. जितने भी हैं, there are 20 plus. In one form or the other, we consume those, right? So those are biological. Fats, fatty acids are part of the body. In the creation, in our food, they are there. Proteins, nucleic acids are the main polymers, which are biopolymers, which are inside the body. But basically, simple 4 molecules, jinki reactivity is that simple. Those who are affected by this simple chemistry, now I will reverse and go in the other direction. 1, 2, and 3. See how many processes inside the body they affect. Look at the protein, for example. Protein cell inside the whole cells. Protein concentration is affecting. Lipids, fatty acids, right? Carbohydrates, we have, as you can see, I have looked at these. They make these molecules, the 5 dotted lines, alliginates, for example, carrageenes, flucoides. All these are, and vitamins, nucleic acids. 4 basic fundamental biopolymers. Inside they create these molecules. Now look at their functions, their methods, their function kahan se hota hai? They create cell disruption ke andar, they have their roles. Mechanical functioning ke andar, chemical reaction, enzymatic reaction, whether look at the physical, and then again enzymatic reaction jo wahan I have listed. But all are these processes which are inside this chemical plant. 4 different types of only biopolymers that I mentioned, and they are affected by enzymatic reactions. Those enzymatic reactions are influenced by simple molecules, simple acids and water inside the water. Water is there is a reason nature has created water, has put water inside the human body, because those solubility of those molecules taking and crossing where cell lines, cell walls, from one type of cell walls to another, to organs, to from plasmas to tissues, it facilitates, right? So, but if you break it down to, they are very simple molecules. So if we understand those, we can look at that and see how they react. So look at the chemistry part, for example. Carbohydrates, saccharides, monosaccharide, disaccharides, polysaccharides. Basically, they are linkers. From oxygen to carbon to hydrogen, they are basically linkers. Those linkers are connecting. So you can understand, if you know the chemical reaction of where these reactions are happening and how their reaction is happening, you can design various biological solutions. You can create artificial carbohydrates. You can create artificial, and there are many now, which are there. But again, it brings down to breaking down to those understanding of the chemical structure, their properties, so that can help you to understand the molecules which already exist, or to design molecules which could be used as substitute. हमने उसका नाम तो काफ़ी सुना है, artificial sugar, right? So कार्बोहाइड्रेट, उसका डिज़ाइन थोड़ा बदल दिया, हमने अपने स्वीटनेस का तो बढ़ा लिया कि उसका मीठा ज़्यादा हो गया. We do not know the negative side of it, and therefore if you go back and look into the literature, there is tons and tons of, there are thousands and thousands of papers which have been published for the positive and negative of the artificial sugar. कि उसके जो sweeteners जो आते हैं, sweeteners के positive and negative, but basically है कि it's a simple carbohydrate molecule which has been modified. But to be able to understand that will be helpful in order to solve the solution, to create a solution and solve the problem. Now, why am I mentioning this? Carbohydrates, I started with carbohydrates, sugar, मीठा खाते हमें taste देता है. Not just the taste, but inside the body, it, the fats converted. You eat a lot of sugar, converted to fat. You create a lot of functions. Go to gym, muscles बनाए, converted to fat, to muscles. But that conversion is, all of these are chemical reactions, right? So, for example, energy function, ATP, ADP, जो conversion है, right? That जो energy machinery जो inside the cell है, ATP, ADP, the conversion that happens. So what happens? This now from cells to tissue. Basically, these are polymers only, they are connected in one or the other form, right? I've shown that picture of cellulose structure on the right. So, but they are connected carbohydrates.
So see, from simple molecules to biopolymers to biomolecules, now they are part of what creates your tissues inside the body. So if we are trying to understand how a drug or a therapy is created, we need to understand what is the function of, what is the makeup of that tissue. If you are interested, अगर आपने wound healing करनी है तो आप अल्जाइमर की तो टेस्ट नहीं करेंगे ना, वुंड हीलिंग का ही टेस्ट ढूंढना पड़ेगा. तो यू हैव टू अंडरस्टैंड हाउ दैट वुंड का जो स्किन है वो बनी कैसे है, उसमें कितने तरह के सेल्स हैं, व्हाट आर द स्ट्रक्चर, हाउ दे आर मेड, व्हाट आर हाउ मेनी डिफरेंट लेयर्स. बट व्हाट दैट ब्रिंग्स डाउन बॉइल्स डाउन टू इज how that structure was created. And then you can create different solutions by creating artificial, these processes. But in terms of energy, again, as you can see, these carbohydrates, these glycogens, for example, that's the storage space for polysaccharides in biological creation. So understanding the functions of carbohydrates, now you can just imagine understanding the function, utility, breaking down of those is basically chemistry, but its application is what is happening inside the animal body. So you can create those creations. Now fats, for example, lipids, right? The fats, fatty acids are basically what? As you look at the linkers, those are fatty acids. Simple. Again, as I was telling you, look at fatty acids, there are only 4 atoms. Carbon, hydrogen, oxygen, actually only 3 atoms. Carbon, hydrogen and oxygen. Now you see how much fatty acid does various different types of function inside the cell, inside the human body. So, but it's basically what? Simple. Carbon, hydrogen, oxygen. So if you can understand the properties, so now you see how I started with the oxygen and H2O, or carbon having four valencies. If we can understand their properties, if we can understand their reactivity, we can better understand the various parts of the cell, various makeup, what is the makeup of the cell, various biopolymers, what those are, how they react, how we can combine, mix and match, what creates a cell, what creates a tissue, what creates a bigger organs, and within those organs, if there are various layers, what each layer is made of. And when you break it down, when you go, they are basically come down to basic atoms or small molecules. So if we can understand those, we can apply. So जितने भी आप टेस्ट अगर आप देखते हैं, टेस्टिंग के जो हैं, whether it's a sugar test, BP का टेस्ट है, जितने भी टेस्ट हैं, diagnostic in general, basically what it is that understanding how these molecules behave under whatever condition is of the assay is. And now this then see, if we combine all of these things together, it now multiple layers. So take glycerol, for example, right? Fatty acid, and I have given you the structure of glycerol. When you have glycerol have 3 different places. Simple hydrocarbon, reactivity with 3 fatty acids. It starts to now build a layer. It starts to bring some creation inside the cell. But going back to, because you are chemist, I am emphasizing again back, simple atoms make molecules, make biomolecules, and then you can, fatty acid itself is a bigger molecule than water and alcohol and super molecule amino acids, right? But when these are coupled together, it now becomes fatty layer. Now fatty layer, various parts of the body has different functions, or makeup might be different. There could be hundreds and different types of fatty layers that were formed. But usko break it down to, they are basically fatty acid. Fatty acids are basically, as you look at the structure, carbon, hydrogen, oxygen. There are only 3 atoms. Only 3 atoms. Now just imagine kitni jagah pe fatty acid human body mein hai. It's only hydrogen, carbon and oxygen. So if we can understand those, you can better understand how the human body is made up, right? So that is where again the advantage as a chemist we have is we can break it down to simple atoms and then start building on top of that, bring it back to the larger molecules. But then again, you need to understand the connectivity, you need to understand. So therefore, it's a lot of fun. It's a lot of fun building the blocks together. Wo apne you know, kids play those Legos and mix and match. These are Legos. These are Legos of the chemists. But it's a lot of fun. But then because now it's not just making molecules, now you slowly start to understand how biological creation is and what and role it may have, any function, any tissue, any cell, any assay, any therapeutics, any models that you create. अगर ड्रग बनाने हैं तो ड्रग बनाने के लिए what models are needed, why those models are needed, it will bring you back to simple atoms. Why? And if you start putting together, your understanding of biological systems slowly start to grow and you will start to connect. And then you say, aha, that is what is happening. This is what basically what chemical biology is. This is what the biological thing is happening, and slowly you start entering into those areas. I am a trained chemist. All my degrees are in chemistry, hardcore chemistry. But frankly, I don't know any chemistry now. I do all disease. I do chemistry. My lab mein hai. I have chemists and PhDs and postdocs and scientists who are only chemists. But 90% of my work in my lab is all biology, because that foundation gives a lot more fun to understand the disease biology. So next time when you hear disease biology, basically kya hai? It's a macrochemistry, right? Agar bade crude sense mein kahe, it's a macrochemistry, chemistry of the macromolecules. But then you have to be able to connect those, connect piece by piece, and then before you know, it becomes a lot of fun. It becomes, and then you want to investigate one after the other, and before you know, you become Sherlock Holmes. You are investigating. You are Sherlock Holmes of the atoms and the molecules. So, but connectivity, bringing back to the topic of this, this lecture, chemical solutions to biological problem. But I'm trying to give you the background, okay, how and why. If I had not, and if you don't think it that way, say, हम तो chemist हैं, हमें biology से क्या लेना देना है? But no, chemist has the advantage जो biologist को नहीं है, जब तक कि वो chemistry ना पढ़े. Why? As I told you, you have the toolbox. You have the toolbox of understanding कि जो मर्जी कह दे DNA, okay, नहीं हम DNA पे काम करें, तो भाई, all right, calm down. You are working on a simply bigger molecule. It's basically a molecule. Now I have to understand उसका क्या है. So therefore, for chemists, as I have seen, and I believe, have the advantage that you have to have that curiosity to ask question and go dig deeper and deeper and to understand. Have the advantage to be able to make sense of whatever the biological is. You may not get it maybe first time, may not second time, maybe it may take you 10 attempts to make. But just try to connect your chemistry understanding, bring your chemistry understanding, your jitna bhi hai, to macromolecules. First time नहीं पता चलेगा, second time नहीं बोलेगा, but bring it down to क्या है. I see. I know exactly now what you're talking. I totally understand कि protein क्या है, RNAs क्या है, DNA क्या है, disease model क्या है, because basically you break it down to simple molecules, and then slowly say, and then you start building it back together. Next time after 10 attempts, when you meet the biologist, you say, Praji रुको, मैं तानू दसना किसना काम कर दे. Then you just switch it. It's the reason I'm trying to tell you is it's a breaking. Any problem, if you look at from the complex side, it looks very big, but you break it down to the problem, to the very small unit, it starts to make sense. Because in order to create a solution, you first have to understand what the problem is, right? You have to understand who the enemy is, what the problem is, before you create a solution. So therefore it becomes much easier for looking from the chemistry angle at the macromolecules, understanding their creation, structure, physiochemical properties, their functions, and then start putting together. And before you know, for example, cholesterol. I mean, I'll throw you BP high hai, sugar high hai, कोलेस्ट्रॉल हो गया है, all these are क्यों हुआ है? You ask, as a chemist, ask the question where, why, how, and then you try to create a balance and understand. Either you want to promote, healthy lifestyle. What definition of healthy may be different. बैठ के दबा के खाना is healthy और exercise करना is also healthy, depending उस individual का देखने का नज़रिया क्या है. But point being, you can bring that chemical solution in understanding the biological problem. So look at few example here. For example, आपने बर्गर खाने हैं, खाइए, जिसमें ऑयल है, फैटी है, लीजिए. आपने सैलड खाना है, पत्ते भी टूट जाएंगे और ये भी टूट जाएगा अंदर जाके. बट ये आपके हैं कि जबान का टेस्ट जबान के नीचे ख़त्म हो जाता है. So what is it that attracts that? But that is what happens. But see, breaking it down to, these are basically macrochemical reactions. So if you look at proteins, kya hai? I have just given few. They are large complex molecules, right? They play critical role different body. Cells के अंदर उनका function है. चाहे वो as an enzyme हो, एक function है, antibodies हो, that's a different function, that the role they play. Then messengers like RNA, they have different structure, and then these are various of their functions that they play inside the cell, inside the tissue, inside the human body. So if you understand, but now उसको solve करने के लिए उसके function क्या है, you take that NADH molecule है, it's a small molecule basically, put together by acid, that makes protein, right? But if you bring it down to the, उसको break it down to the small brick, it's an amino acid, right? So understanding, so now knowing that that's what it is, you can come up with what should be the way to test those, create assays for those, create applications for those, because now you can test protein hai, to which protein is there, what their functions are. You can create models, you can create assays, various things can be done. So this is another way of showing you. This is all the things from your breakfast to whatever you eat and whenever you eat. But those are the 20 naturally occurring amino acids. Now look at the simplified structure. One side is amine, other side is acid. And then you can have different linkers, you can have different R groups. That basically gives to. Now, putting together these 20 bricks in different combination creates the variety, thousands of different proteins. Permutation combination, you can just imagine how many you can create, whether peptides or proteins. And depending on what is the makeup of the protein, right, different function, its application will be different. So I like to, how many of you know the genome structure जो हुआ, the human genome project, which was NIH 20 years ago plus. So the genetic analysis, जो human genome versus dog, pig, elephant, varieties of these things that have happened. A big chunk, a large percentage, in many cases depending on 90% plus, human genome is similar to these animals. It's just a small variation. Obviously that permutation combination, okay, one leads to human, other leads to pig or dog, or so. But basically to understand what gene makeup is, what protein makeup is, whatever that is, it breaks it down to that. So as many chemical understanding we can create, you can bring those chemical solutions to the protein problems. And I'll just skip this. So these are basically, as I was telling you, these are basically reactions. Water comes out, as you see, two amino acids, glycine and alanine mix, it makes a peptide. We'll keep on mixing, it will convert into a protein. It will become a protein, and where they react, how they react, will create. And now that creates various protein structures, whether they are primary structures, they are secondary structure, tertiary structure, quaternary structures, depending on how these molecules are reacting or creating, new structures are formed. Based on what structures are formed, their functions inside the cell, their function inside the tissue, their function inside the human body varies. But in order to understand which protein is doing what, you have to be able to analyze that. In order to analyze that, you have to understand what the chemical makeup is. In order to understand chemical makeup, you simply have to understand what the chemical solution, chemical structure of that is. So you see, from diagnostic to therapeutic, if you want to create any new entity or even to understand what are the existing sciences, interesting assays are, interesting diagnostic tools are, or therapeutic solutions are, basically they are chemical makeup in different ways, and if you put those things together, your understanding of the human body, the human disease, they are a utility of various assays, models, tools that you see changes. And same way, you know, enzymatic reactions kya hai? Human body ke andar there are thousands and thousands of enzymes. Basically what is happening is a simple chemical reaction. A structure will come, bind to an enzyme, forms a complex, they separate, a product is formed. Enzymes are basically the catalyst. Nature's catalysts are enzymes, right? And so, or they help in synthesize different things. So again, knowing these things can help understand, put the pieces together. Now look at, for example, you know, your sugar molecule connected to a base, connected to a phosphate, it forms a nucleotide, right? There are so many of those inside the human body. But unka basic foundations are simple, these small molecules. So if you, before you know, it starts to create and start to make sense, right? RNA, ribose, nucleic acid, break it down to what it comes down to. RNA jo hai, sugars ka role, if you understand the roles of sugars, if you understand the roles of the bases, if you understand how many of these strands are together, you start to bring what it makes up, what is the bigger macro synthesis, and basically then you start to understand, you know, a lot more about the complex human body. Any question? Yes. I'm sorry, say it again. किसका? मिल्क का, ओके, न्यूट्रल. हाँ, पनीर बनता है. यही तो क्रिएशन का कमाल है कि वो ब्रेक करता है तो जल्दी से उसको gets into different cells. That's why I said this is the most complex and the most beautiful creation, and we're still trying to understand because it simply breaks it down. वहाँ जाके प्रॉब्लम इसलिए नहीं करता है क्योंकि वहाँ जाके बेसिकली क्या है, वो कैसीन है, you know, brings break down कैसीन है, कैसीन के functions है, right? We are in Panjab, दूध पियो, they are aired everywhere. Why? Because it breaks down, it gives you the protein, separates the protein. Now I'll tell you another thing why in most diets, for example in the Western diet, there is a combination of omelette and orange juice. जो मैंने अब तक का लेक्चर दिया है, do you understand that उसका एडवांटेज क्या है? Omelette and orange juice is a very commonly used combo. Why? I just gave you the solution. हमें चाहिए क्या होता है? Omelette is what? Rich in protein. Orange juice is citric acid. It breaks down to give you amino acid and energy very quickly. हमारा जो फूड है उसमें ज़्यादा शुगर है. We are carbohydrate enjoy करने वाले हैं. So उसमें शुगर बेस ज़्यादा है. So you're right. It's a good question कि stomach में जाके भी तो टूटेगा, but stomach में जाके वो टूटेगा. It breaks it down to molecules which are needed for the human body, जो energy देते हैं. Water separate हो जाएगा because protein, you know, it's a mixture of many molecules, but casein जो उसका protein है, and then water जो milk है, right? So casein gives immediately protein, so that is needed. तो टूटे तो अच्छा है कि टूटता है वो, but that's what it does. Same way any other food, for example. But the one that, for example, now ATP, as I've been telling you a few times during the day, ATP जो है, energy जो है, plant जो है, human cell के अंदर जो है, right? वो है क्या basically? Look at that, phosphate, ribose, amino acid, carbohydrate, सबकी chain है. वो उसको break down करता है energy, energy जो है, there are exothermic reaction, endothermic reactions. So body is again, somewhere it's an endothermic reaction which is happening, somewhere it's an exothermic reaction. This is an exothermic reaction which is happening because it breaks it down, energy generate होती है, that gives the boost. Right? सुबह उठ के आलस आता है. The moment you eat glucose, थोड़ा भी आप मीठा कुछ खाते हैं, कुछ लेते हैं, you stand up. Right? क्या हुआ कि glucose अंदर गया, the carbohydrate अंदर गया, it broke it down. Right? इसका energy generate हुआ, you feel like okay, let's get to work. So basically, if you look at this, there are these understanding of how these biopolymers or the biomolecules are made up of, what is their fundamental chemical units, understanding the roles of those fundamental units, their structure, their physiochemical properties, and the functional role that they play. You can come up, now you can see, I hope you have a better appreciation of how chemical based understanding foundation, more and more you make it stronger and stronger, it will help you to better understand the biological molecules, their functions. That leads to understanding hopefully someday disease जो है, disease, what is disease? Basically some malfunction हो गया, जो proteins हैं, जो भी चीज़ है उसका malfunction हो गया, उसको समझने के लिए, diagnose करने के लिए आप you take your car to the mechanic has to first understand कि कहाँ पे break, जो भी चीज़ टूटी है या fix करनी है, in order to understand उसको पता होना चाहिए कि उसको fix कैसे करना, उसको पहचानना कैसे. यही है कि ये कुदरत की बनाई machine is a little bit more complex. तो उसे समझने के लिए the more we understand, so we can provide those chemical solutions for these biological problems. So you see, now I started with the slide that I showed you. Now if you see this, from atoms to molecules, they create these things, right? Where they are proteins, they are fatty acids, the carbohydrates, nucleic acid. This is the creation which is inside the cells. These cells then make up your tissues. Tissues make up your organs. Depending on whether primary protein or secondary structure, or tertiary structure, quaternary structure, each one has its role to play. Depending on that, उसके अंदर muscles बनते हैं, fats बनते हैं, जो भी है. But all of that leads to this complex ocean we call cell. It's all these things are happening inside nature's creation. So you see again, the reason I wanted to show you this was, it starts from chemistry. Simply understanding the fundamental of molecules, simple molecules that are relevant to whatever problem that you're interested to solve, whatever thing that you're interested to learn about, to understand related to biological, in this case, let's say for example, human body or human diseases. It will bring you down to this side. And brick by brick, you can develop that understanding and then can use that understanding to create solution where there is a diagnostic of a disease or hopefully create therapies for the disease. So just this is, bringing, that's why I gave the title to this, bringing chemical solutions to biological problems. Any question? If not, we are done for the day. All right, so that's all. I didn't want to bombard. I mean, this is, I hope whatever I'm trying to share with you is making sense. If not, do not hesitate. I actually like to interact, because if it's a monotonous, if I'm doing only one direction, if it is not being received the same way, it is not landing on your side the same way, then purpose defeated. And I don't want that the time that we have together to be wasted. हो सकता है मेरे समझाने में you need more example, I can come up with more example. But my goal is that you understand whatever I'm trying to say, at least what I'm trying to convey, you understand that. If not, absolutely no problem, raise hand and just tell, okay, please repeat. मेरे को झपकी आ गई थी, ऊपर से निकल गया. Don't worry, I don't mean by that. आ भी सकती है, but what I mean is that don't hesitate, ask question, and if you want me to repeat again, no problem, I can go over again. If you want any solution, example, if you need any suggestion, like I said, this folder is for you guys, and I will give Dr. Singh this folder, but I will also give him the soft version, so you will have all those copies. Then try to go through, and you'll see that the things which I'm telling, you'll find some relevance to papers which are relevant to the, I have, I don't know, maybe around over 20, maybe 25 different review articles which are relevant to all the topics that I'll be covering. All right, thank you very much. If nothing else, enjoy the evening. We'll see you in the morning.